Good morning, ladies and gentlemen. My name is Nuri Azbar, chairman of the workshop. Use the channel as you wish, either English or Turkish. Both is available right now. I have the pleasure and honor to welcome you all to our international workshop on enhanced biogas production and recent innovations. I would like to thank all our participants for your interest, and a special thanks to our distinguished speakers and moderators for their valuable contributions. Last but not least, another special thanks to our sponsors, of course. Without them, we cannot realize this workshop. Of course, Dalia Tour, especially Mr. Ekrem Gülay, deserves a great appreciation for their excellent organizational management of this workshop. As you all know, climate change is now considered as climate crisis due to the ever-increasing fossil fuel-based carbon dioxide emissions all over the world. Both Paris Agreement and European Green Deal aim to realize a carbon-neutral world until 2050. In my opinion, not a carbon-neutral but negative carbon world should be realized to cope with this climate crisis. In this manner, CCU, which is carbon capture and utilization technologies, should also be developed as fast as possible. And I will try to mention this in my presentation next day. In order to be able to cope with the climate crisis, use of renewable energies is crucial and indispensable. Amongst these renewable alternatives, use of biogas technologies exploiting waste biomass seems to be very promising. The solution is to switch our point of view in regards to disposal of organic wastes.
Organik atıkların bertarafına ve atık bertarafına önem vererek bu konuda büyük adımlar atabiliyoruz.
For example, at least in Germany, there are more than 2,000 biogas plants. The estimated number in Europe is over 18,000. I'm not even counting the ones available in China, India, and other countries. Even though biogas technologies are well known, there is still significant room for development and enhancement in process technology in a way to increase biogas production efficiencies and reducing hydraulic retention times, reactor volumes, and other attempts that we will discuss for the next 2 days. Those are the pre-treatment and other attempts. I strongly believe that our workshop on enhancement and recent innovations in biogas technologies is quite timely and meaningful. I wish you all a successful and fruitful meeting during these 3 days. For the first 2 days, we will be having the scientific and technical discussions, and the third day, we will have some round table meeting with the potential companies and the potential customers. This is going to be a matchmaking meeting in the third day. I also wish that you all stay safe and healthy in these critical days due to the COVID pandemic. As you all know, we have been experiencing just like a Hollywood scenario for the last one and a half year, and I hope that you're all safe in these weird days. By that, I now invite our next speaker, Mr. Tolga Ilçin, who is General Secretary of Association for Bioenergy, in Turkish is Bioder, and I'm inviting him for his opening speech, and I thank him for his contribution. Kind regards. Mr. Tolga Ilçin, the stand is yours. Please go ahead. Thank you for your contribution.
Teşekkür ederim hocam. Değerli hocam ve değerli katılımcılara bu güzide çalıştayda ve sektörümüzün bu güçlü açıkçası bugün. Bu sunumu bugün Başkanımız İlker Bey yapacaktı ama küçük bir rahatsızlığı nedeniyle katılamadı. Çok selamlarını iletti her birinize. O yüzden bugün sizlerle biyokütle sektörü ve Bioder hakkında küçük bir bilgilendirme sunumu yapmak istiyorum. Özellikle biyokütle yönetimi bu çalıştaya da konu olduğu üzere ülkemizde giderek popülerleşen bir konu ve popülerleştikçe çözülmesi gereken sorunlar bir mertebe daha su yüzüne çıkmaya başladı ve sorunlar ve ihtiyaçların çözülmesi sektörün büyümesiyle büyümemesi arasındaki ince bir çizgi haline geldi. Bu anlamda çok önemsiyorum bugün burada bulunmayı ve bu sorunlara önümüzdeki dönemde de çözüm getirebilmeyi. Nacizane bizim görüşümüz, derneğimizin çabaları 2014 yılında kuruldu Biyoder ve 2014'ten bu yana sektörün gelişimi ve paydaşların sorunlarıyla ilgili bir sivil toplum kuruluşu olarak faaliyet gösterme çabası içerisindeyiz. Burada hem sektör oyuncularına hem de bakanlıktaki bizim çözüm ortağımız paydaşlarla bir köprü vazifesi görmeyi arzu ediyoruz. Buradaki en büyük sorunsal sektörün mahiyetinin iyi tanımlanması gerektiğine inanıyoruz. Atık yönetimi ve atığın geri kazanımı nosyonuyla çalışan bir sektörün birden fazla bakanlıkla, birden fazla kurumla, birden fazla paydaşla çalıştığı ve bunların aslında çözüm odaklı bir çalışma, gönüllü bir çalışma felsefesiyle yatırım yapıldığı bir sektörden bahsediyoruz. Dolayısıyla atık yönetimi ve biyokütle kavramlarını bir arada bulundurmak bir nebze olsun konunun daha iyi anlaşılmasına fayda sağlayacağına inanıyorum. Bugüne kadarki biyokütle çalışmalarında biyokütleyi hep enerji olarak algılamakla beraber aslında biyokütle yönetimi bir atık yönetimi ve operasyonudur, bir geri kazanımıdır. Farklı fraksiyonları olabilir. Fakat bu fraksiyonlar sonuçta toprağa dönen organik maddeye kadar giden bir atık yönetimidir. Ve bunu iyi yaptığınız takdirde döngüsel ekonomi kavramını tam yerine oturtmuş oluyorsunuz. Kısa bir sunum var. İsterseniz onu paylaşayım. Hem derneğimizi hem de sektörü anlatmış olurum. Oradan sonra çok sözü uzatmadan diğer konuşmacılara da fırsat vermek istiyorum. Şimdi biyokütle yönetimi, ekrana yansıdı sanırım. Temel anlamıyla yenilebilir enerji kavramı içerisinde bir alt çatıdan oluşuyor. Biz atık yönetimi yapıyoruz ama fosil yakıt kullanmadan bir enerji üretme potansiyelimiz var. Farklı kaynaklardan besleniyor bu sektör. Ve bunun en büyük avantajı da atmosfere saldığımız karbon salınımını minimize eden bir enerji yönetimi usulü. Ve bu iç kaynaklardan yönetilebilen bir sektör. Yenilenebilir enerji kullanımında dışa bağımlılığı ortadan kaldırabilen bir üretim metodu. Özellikle iklim krizini yaşadığımız bu günlerde karbon emisyonlarının öne arz ettiği ve tüm enerji politikalarının tekrar gözden geçirildiği bu günlerde biyokütle yönetimi sadece enerji üretimi açısından değil, enerjinin alternatif maliyetlerini de engellemesi açısından da önem arz edeceğini düşünüyoruz. Atığın serbest halde ortada bırakılmasıyla oluşabilecek negatif sonuçlar bu sektörün yaptığı çabalarla tekrardan hem bir ürüne, bir enerjiye dönüşebildiği gibi aynı zamanda atmosfere salacağımız metan, karbondioksit gibi emisyonunu da önüne geçmekte. Peki bu sektörde neler işleniyor? Bir biyokütle sektöründen hangi çıktılar var elimizde? Bu tabloda elektrik, gaz, biyoetanol, sıcak su, buhar gibi enerji kaynaklarından bahsediyoruz. Ama bunlar sadece başlı başına bir enerji kaynağı olarak düşünülmemeli. Bunların sonunda tesislerin dışarısında bir de organik katı madde çıkıyor. Sıvı fermente ürün çıkıyor. 2019 senesinde FAO'nun hazırladığı bir raporda görüyoruz ki topraklar organik karbon açısından iyi bir karbon yutağı. Atmosferde serbest salınan ve karbon bazlı yakıtlarla atmosfere salınan karbondioksidi tekrar toprağa döndürebildiğimiz takdirde iklim değişikliğinde iyi bir enstrüman olarak kullanabileceğinden bahsediyor FAO. Bu minvalde bizim içinde bulunduğumuz bu sektörün de bu çalışmalara önemli katkı sunduğunu düşünüyorum. Organik atık yönetiminde bir disiplindir biyokütle yönetimi aslında. Alternatif bir çözüm olarak görüyoruz ama serbest olarak dolaşan atıkların bir formasyonla toplanıp, bir lojistik yönetimiyle işlenip, farklı disiplinlerle enerji veya katı sıvı ürünlere dönüştürülüp tekrardan toprağa ve enerji olarak son tüketiciye ulaştıran bir ara dişlidir bu sistemde. Doğanın yıllar içerisinde işleyip yavaş yavaş yönettiği bu süreci biz belli tekniklerle farklı disiplinlerde hızlandırarak yönetiyoruz aslında bu biyokütle yönetimi içerisinde. Farklı atıklar bizim ham madde kaynağımız. Burada atık ve artık kavramı çok önemli. Artık dediğimiz şey geri dönüştürülebilir. Bu çalıştayda bu kavramın da yerine oturacağını düşünüyorum. Doğru artıkları tanımlarsak atığın atılması gereken, artığın işlenmesi gereken ve nasıl işlenmesi gerektiğini altını çizeceğimizi düşünüyorum. Biyokütle enerjisi farklı biyometrizasyon tesisleriyle elde edilebildiği gibi landfill gibi, yakma gibi farklı formlarda da enerjiye dönüştürülmektedir. Burada dediğimiz gibi az önce atık karakterizasyonunun ne olduğu çok önemli bizim için. Avantajları nelerdir bu sektörün? Bir kere sürdürülebilir bir sektördür. Eğer artık kaynağınız ya da ham madde kaynağınız sınırsızsa sürekli olarak enerji üretebilirsiniz ve bunu dışa bağımlı olmadan yapabilirsiniz. Özellikle enerji arz güvenliği için çok önemli bir sektördür. Kendi kaynaklarınızla enerji üretebilirsiniz. Ve buradan elde edilen ürünleri tarımsal üretime aktardığınız için gıda arz güvenliği içinde hayati öneme sahip olduğunu düşünüyorum bu sektörün. İlk konuşmamın başlangıcında da belirttiğim gibi Biyodel 2014 yılından beri faaliyette ve bünyesinde bulunan mühendislik firmaları, işletmeci firmalar, tedarikçi firmalar, danışman firmalarla sektörün sorunlarına bir yol haritası çizme arzusuyla faaliyetlerine devam ediyor.
Of our sector, and we are helping our clients with
önümüzdeki dönemde özellikle Tarım 4.0, iklim krizi ya da biyokütle yönetimi gibi konular önümüzdeyken sorunların masaya yatırılıp ne minvalde çözüleceğine karar vermemiz gerekiyor. Belki bu çalışmanın takip eden yıllarda da bir kontrol noktası gibi tekrarlanıp bize bir yol haritası vermesi ve her sene nerede olduğumuzu, geçmiş yıllarda ne yaptığımızı sürekli takip etmemiz bizim sektörümüzün ve bizlerin de yararına olacağını düşünüyorum. Ben sözü fazla uzatmadan diğer değerli katılımcılara da teşekkür ediyorum burada bulundukları için. Zevkle dinleyeceğim. Güzel bir çalıştay olmasını temenni ediyorum. Teşekkür ediyorum Nuri Hocam.
Thank you very much, Mr. Elchin, for your valuable contribution. And I also thank you for your additional clarifications on some critical definitions, especially on waste definition. From the governmental legislation's point of view, waste is a big problematic issue. But as long as we call it waste, we're in the sink of problem. But if we change our point of view from waste to value, you know, that's where the solution starts. That's, I believe, from the bottom of my heart. And the industrial people I know and the NGOs like yours, and we will be having some other president of the other NGOs, non-governmental organizations, and I really value
destekleriyle bu konunun üstesinden geleceğimizi düşünüyorum.
the cooperation between the NGOs and the government, so that this is for the beneficiary of our country and for our world. And you know that we will be mentioning in the next 2 days in regards to the Green Deal, which has quite a bit critical issues for the industry. There are so many limitations and the risk for our international business. So we will be also discussing that, and I really thank you for your existence for the workshop. Thank you for your contribution. So we will continue with Mr. Altan Denizsel, another important association in Turkey. It's called Association for Development of Biogas Investments in Turkey. It's Biogas there. Mr. Denizsel, I also thank you for your contribution, and stand is yours. Please go ahead and let's have your opening speech. Thank you.
Merhaba hepinize. Böyle bir çalışma düzenlediğiniz için ilk önce hepinize teşekkür etmek isterim. Her ne kadar biraz geç kaldı, çünkü yeni eklem çıktı. Bu saatten sonra pek fazla da bizim sektöre yatırım yapacak adam da kalmayacak bu 54 kuruşlarla da. Belki yani son şeyleri kaçırdıklarımızı yakalayabiliriz diye düşünüyorum. Tam öğrenmeye başlamıştık, tam bir şeyler yapmaya başlamıştık. ama önümüz kesilmiş vaziyette. Bundan sonra da ben çok fazla umutlu değilim. Özellikle biyogaz sektöründen, yani ıslak fermentasyondan çok fazla umutlu değilim. Diğer sektörler için konuşamam. Bir de tabii Türkiye'de genelde Biyogaz Derneği olarak biz 2009'larda, 2008'lerde kurulduğumuzda ortada biyogaz olmadığı için derneğimizin ismini mecburen Biyogaz Geliştirme Derneği olarak kurduyduk. Çünkü biyogaz yatırımcısı ortada yoktu. Ve öyle devam ediyoruz. Türkiye'deki bu işlerin daha iyi olması için bazen duvara konuşuyoruz, bazen işte kendi kendimize konuşuyoruz. 100 tane anlatıyoruz, bir tane uygulanıyor gibi derken bugünlere kadar geldik. Tabii bizim amacımız hem Türkiye'de biyogazın, ıslak fermentasyonun gelişmesini sağlamak. Bu arada da yurt dışı know-how'lara çok fazla para ödemeden bu işi nasıl Türk mühendisleriyle, Türk
into
akademi
Turkey without paying money to international.
koştuk senelerden beri. Zaten son zamanlarda da dikkat ederseniz derneğimizin üye potansiyelinde ağırlıklı ekipman tedarikçileri oluşmaya başladı. Kendilerini daha iyi ifade etmek açısından. Ve diğer firmalar da referans almaya başladılar bunu. Tabii Türkiye'de en büyük olay kavram karmaşası. Yani biyokütle. Biyokütle dediğiniz zaman zaten genelde herkes yakma anlıyor. Ama biyokütle bunun bir çatı yapısıdır. Tolga kardeşim zaten e, çoğu şeyi detaylı olarak anlattı. Ben aynı sorunları burada tartışmayacağım, ortaya koymayacağım. Ama yani biyokütle dediğimiz zaman da yani bunun bir ıslak fermentasyonu var, kuru fermentasyonu var, landfill'i var, pirolizi var, gazifikasyonu var, yakması var, plazma gazifikasyonu var. Bunların hepsi ayrı ayrı uygulamalar yapılması gerekiyor. Türkiye'de de yanlış uygulamalar, yani gazifikasyon yapacak yerde yakıyorlar. Yakacakları yerde piroliz yapıyorlar. Öteki tarafta işte mesela şimdi yeni bir moda çıktı. İşte çöp atıklarındaki organikleri ayıracaksınız. Bir ıslak fermentasyon, kuru fermentasyon yapacaksınız. Biyometanizasyon deniyor. Kurulan tesislerin hiçbiri adam gibi çalışmıyor. Saçma sapan şeylere para ödeniyor. Burada tabii ben en büyük hatayı Çevre Bakanlığı'nda görüyorum. Yani Çevre Bakanlığı üniversitelerle ve de sivil toplum kuruluşlarıyla bir araya gelerek bazı şeylerin artık ismini koyması gerekiyor. Yani ne yakılır, ne gazifiye edilir, ne landfill olması gerekiyor. Bütün bunların ayrışması gerekiyor. Artık yani bir de bunlar için arge yapmaya da gerek yok. Dünya artık bunu biliyor artık. Yani yığına örneği var. Yani şimdi çöpü yakacaksınız. Yakacaksınız da çıkan ısıyı ne yapacaksınız? Başınıza bela. Avrupa yapıyor. Yapıyor da adam Kopenhag'ı ısıtıyor. Adam Amsterdam'ı ısıtıyor 40 seneden beri. Siz atık ısıyı kullanamazsanız tesis verimli olmuyor. Hem ısı hem elektrik parasını alamazsa. Yakıyorsunuz, filtrasyon kullanmıyorsunuz. Daha da tarımsal atıkların yakılmaması gerekiyor. Bunların gazifikasyon yapılıp biyoçar denen de ikinci bir maddeye girmesi gerekiyor. Tabii burada gene bakanlık bunları bir standart haline getirmesi gerekiyor. Biyogaz yapıyorsunuz, Tarım Bakanlığı çıkan sıvıyı gübredir diyor, tarlalarınızı, hayvansalıkları dökün diyor. Döküyorsunuz, ertesi gün Çevre Bakanlığı geliyor, ceza yazıyor. Kimin muhatap olduğu belli değil bizim sektörümüzde. Yani güneş, rüzgar gibi bütün bu diğer yenilenebilir enerjilerin tek sahibi var, Enerji Bakanlığı, ama bizim sahibimiz yok. Yani biz Tarım Bakanlığı bağlıyız. Çevre Bakanlığı'ndan bağlıyız. Enerji Bakanlığı'ndan bağlıyız. Yani Enerji Bakanlığı tamam, onlar da haklı, yani ilelebet yüksek fiyatlarla bu işi destekleme şansları da yok. Onlara da bir şey demiyorum. Çünkü insanların da ucuz enerjiye ulaşması gerekiyor baktığınız zaman. Ama tamam da yani biz atıkçıyız, yani günde şimdi şu anda bakın 220 megawattlık bir biyogaz tesisi oldu. Ortalama günde biz burada tesis başına atıkları günde 300 ton, 500 ton olarak hesap ederseniz çok büyük bir rakamı biz her gün sistemin içinde yeniden kullanılabilir hale getirmeye çalışıyoruz. Yani Türkiye'de 500 bin ton günlük inek atığı var. 40-50 bin ton tavuk atığı var. 100 bin ton şehir atığı var ki diğerlerini sayamıyorum. Yani mezba atıklarından, sera atıklarından, hal atıklarından gibileri de koyarsanız milyonlarca ton atık var. Bunları bir gün işlememeniz demek o zamanı telafi edememeniz demek. Yani yerine ya kapasite artıracaksınız da bir günlük kapasite artıramayacağınıza göre sürekli bunun her gün aynı disiplinde yok edilmesi gerekiyor. Biz şimdi bunları yapıyoruz. Çevre Bakanlığı demesi gerekiyor ki al bak yeraltı suyu kirlenmesinin etkisini ortadan azaltıyorsunuz. Size şu bir su bonusu vereyim. Ne bileyim hava temizliyorsunuz, metan gazı yapmıyorsunuz. Bu karbon teşviği vereyim. Tarım Bakanlığı desin ki siz fermente gübre yapıyorsunuz burada. Herkes organik diyor da organik beslenirse organik olur. Ama fermente gübre çok faydalı bir gübre. Siz fermente gübre yapıyorsunuz. Size bir destek verelim. Şimdi organomineral denen bir şey var. Ya ilk önce fermente gübreyi yap da yani organomineral gübreyi sonra yaparsın yani. Türkiye'de biyogazdan çıkan organomineral gübreyi yapabilecek 2 tane tesis vardı zaten. Onlar da tam kapasiteyle yeni çalışmaya başlayacaklar. Yani bize bir şeylerin verilmesi gerekiyor. Biz benim sektörümdeki yatırımcıların hepsi yan gelip yatıcılar değil yani yatırımcılar. Her gün sabahleyin aynı şekilde başlıyorlar. Her gün aynı disipline o pislikler toplanacak. Sahaya gelecek. İşlenecek. Bir de bize ne girerse o çıkıyor. Ne girerse o çıkarsa bir de onu yöneteceksiniz. Şimdi onu yönetmeye kalkıyorsunuz. Onun için de ek teşvikleri verilmesi gerekiyor. Gidiyorsunuz bankaya. Gübreyi satabiliyor musunuz? Şimdi enerjiyi satıyorsunuz ya. Devlet alıyor. Ama gübrede bir problem var. Banka kredi vermiyor gübre işleme tesisine. Çünkü neden? Öyle alım garantisi yok ki bir taraftan. Öyle olunca da tabii ikinci tarafa doğru geçemiyorsun. Bir de bunun başka bir yolu daha var. Ya 5 megawatt tesisi, gübre işleme tesisi ya da şey kurarsınız da ya 1 megawattlık, 1,5 megawatt tesise yaptığınız yatırımın yarısı kadar da bir gübre işleme tesisi kuramazsınız yani. Şimdi 1 megawattlık, 1,5 megawatt tesise 600-700 bin dolar daha, euro daha gübre işleme tesisi kurma şansınız yok. Çünkü hem kredi alamıyorsunuz. Bunların işlenmesiyle ilgili problem var. Başka bir destek yok. Biz bir de yani çok pislik içinde uğraşıyoruz. Bunu hiç kimse görmüyor. Bizde salmonella var, bizde brusella var, bizde tüberküloz var. Yani baktığınız zaman başka ne hastalıklar vardır. Onu doktor ve veteriner arkadaşlar da benden çok daha iyi bilirler. Bir de biz bunlarla yani her türlü kontamine hastalıklarla mücadele ediyoruz, topluyoruz. Çiftlik sahibinin ağız kokusunu çekiyorsun, üstüne para ödüyorsun. Halbuki dünyanın her yerinde...
Çok özür dilerim. Programın sürekliliği için toparlayabilir miyiz acaba?
Toparlayabiliriz, tamam. Ya bunların konuşulacak çok şey var sektörümüzün dertleri açısından. Ama burada şunu önemsiyorum. Bu programda biz tecrübe sahibi olmak istiyoruz. Neden? Çünkü reçete düzenleyen adam yok Türkiye'de. Biz mekanikçi, elektrikçi ve bütün herkesi biyogaz tesislerine adapte edebiliyoruz. Ama doğru reçete hazırlayan, biyoteknikçi, biyokimyacı, hangi ismi verebileceksek bunun eksikliği var Türkiye'de. Bizim üniversitelerden ve de bu tür çalışmalardan en büyük isteğimiz bu insanları piyasaya bilgili halde sunabilecek bir eğitim programı. Bu Ege Üniversitesi'nde sizin kürsüde de olabilir hocam. Biyokütle merkezinde de olabilir Ege'de. Yani bizim adam eksikliğimizin tek noktası reçeteyi doğru hazırlayacak kişiyi bulamamamızdan kaynaklanıyor. Herkes çok biliyor Türkiye'de. Herkes biyogaz uzmanı. Herkes biyogazın uzmanı olmaz. Ya biyogaz 4 tane disiplini var. 4 tane disiplini. Bu 4 disiplini tek bir adamın bilmesi mümkün değil. O yüzden bu akademik çalışmaları hızlandırabilirsek belki eleman bulma konusunda da tesis sahipleri bir yol bulabilecekler. Çünkü onlar da bilmiyorlar nasıl yapacaklarını. Böyle bir programa da öne ayak olduğunuz için çok teşekkür ediyoruz sektör paydaşları açısından. İnşallah yüz yüze daha aktif çalışmaları şu koronavirüs geçtikten sonra beraberce yapabiliriz. O günleri özlüyoruz. İnşallah başarılı bir program olarak devam eder 3 gün boyunca.
Well, Mr. Denis, thank you very much for your really interesting contributions. You have drawn attention to very distressful, problematic issues in regards to the sector's problem. You're right, and I agree that the new legislation for renewable incentives it is almost reduced by 40%. Now it used to be $13.3 per kilowatt hours, but now I think it's on the average 7.5. You're right, but I think our workshop, it kicks in the right time right now, because what I believe that we have even more motivation now to enhance and increase these efficiencies in these biogas plants, because we have to find out or figure out a way to produce more energy with less expensive materials, or less operational or capex and opex. This is where the universities kicks in, together with the industry, together with the government. And I thank you for your suggestion that the industry is a crucial part of these pillars. That's the university, industry, and the government have to work all together. But I disagree with your opinion that there is nothing else to discover anymore. The main point of this workshop is to underline that there are still another maybe 100 years long research for biogas plants. As you remember, or as you already know, that we have retention times around starting from 15 days, sometimes goes up to 60 days. Why not we drop down or reduce this retention time down to a few days? Why not we increase the volumetric energy production rates 3 times more? So this is where university still kicks in, and I strongly believe that Turkish scientists are quite a bit strong player in this sector, not the Europeans only. But I agree with your all your suggestions. Industry is suffering already, but now these new incentives with the renewable legislation, it's in Turkish, it's YEKTEM, unfortunately they are reduced. As you already mentioned, that we already had only 60 or around 65 biogas plants working in Turkey, and our potential is around 3,000. And together with this now reduced incentives, of course, the investors will be thinking about with the new investments. But there is a chance that we can stress on the biomass management part, as Mr. Ilchin already underlined. That is the organomineral fertilizer, because we are supposedly or hypothetically agricultural country, but we have been using millions of chemical fertilizers. So the biomass coming out of the biogas plants that's digested is a very, very good potential for the organic material for our lands, which are already suffering from the organic material. It's only 0.5 or 1% organic material. It's supposed to be over 5%. So, but thank you for your valuable contributions. Since we have limited time, I'd like to continue with Mr. Hüseyin Vatansever, another important association in Turkey that's called Association for Energy Industrialists and Businessmen. In Turkish, it's ENSİA. Mr. Vatansever, I also thank you for your valuable contribution. Stand is yours. Go ahead, please.
Teşekkür ederim sevgili Nuri Hocam. Sayın Çalıştay Başkanım, kıymetli akademisyenler, hepinizi Enerji Sanayicileri ve İş Adamları Derneği Yönetim Kurulu adına sevgi ve saygıyla selamlıyorum. Derneğimizin akademik üyeleri arasında yer alan ve ülkemizde biyoketli enerjisi alanında en önemli otoritelerden biri olan Profesör Doktor Nuri Azbar'ın önderliğinde düzenlenen etkin biyogaz üretimi ve güncel yenilikler çalıştayının Türkiye'nin yenilenebilir enerji sektörüne olumlu katkılar sağlamasını diliyoruz. Türkiye yenilenebilir enerji ve enerji verimliliği alanında ekipman, teknoloji ve proje üreten uluslararası bir merkeze dönüştürmek vizyonu ile 2016 yılında çalışmalarına başlayan ENSİA, bugün ülkemizin farklı kentlerinden ailemize katılan ve alanlarında ülkemizin saygın firmalar arasında yer alan 40 kurumsal üyemiz ve 14 bireysel üyemiz ve aynı zamanda 12 akademik üyemiz ile sektörümüz için değer yaratmaktadır. Derneğimiz ülkemizin en açık yenilenebilir enerji kaynağı olan biyokütleye çok büyük önem vermektedir. Türkiye Elektrik İletim AŞ verilerine göre ülkemizin 2020 yılı sonu itibariyle ulaştığı yaklaşık 96.000 megawatt elektrik enerjisi kurulu gücü içinde biyokütle enerjisinin payı maalesef sadece 1115 megawatt düzeyindedir. Toplam kurulu güç içerisinde sadece %1.1'lik bir orana karşılık gelen biyokütle enerjisinin Türkiye'nin yerli ve yenilenebilir enerji kaynakları arasında adeta dokunulmamış hazine görüntüsü verdiğini düşünmekteyiz. Enerji ve Tabii Kaynaklar Bakanlığı tarafından yenilenen ve kısa adı BEPA olan biyokütle enerjisi potansiyeli atlasının da bu durumu tüm çıplaklığıyla ortaya koyduğunu görmekteyiz. BEPA'ya göre Türkiye'nin hayvan ve bitki atıkları, evsel atıklar ve orman atıklarından kaynaklanan elektrik üretim potansiyelinin yılda 14.6 milyon ton eşdeğer petrole karşılık gelirken, ülkemizin birincil enerji arzının 2020 yılın itibariyle 150 milyon TEP seviyesinde olduğunu da görüyoruz. Türkiye'nin tamamıyla yenilenebilir yerli bir enerji kaynağı olarak daha fazla farkındalığa ihtiyaç duyduğuna sürekli olarak dikkat çekiyoruz biyokütle enerjisinin. Bu alandaki enerji potansiyelimizin ise bugünkü değerinin en az 10 katı olduğu tüm kamu otoriteleri yayınları ve bilimsel araştırmalarda ortaya çıkmaktadır. Kıymetli katılımcılar, yerel yönetimlerimiz başta olmak üzere tüm paydaşların biyokütle enerjisine yatırım eksikliği nedeniyle kullanamadığımız büyük bir potansiyele dikkat etmesi gerektiğini söylemek istiyorum. Türkiye'de 2020 sonu itibariyle irili ufaklı 275 biyokütle kaynaklı elektrik üretim tesisi bulunmaktadır. Tüm evsel ve hayvansal atıklarımızın müthiş bir enerji kaynağı olduğunu bilmemiz gerekiyor. Yerel yönetimlerin evsel atıkları kaynağında ayrıştırdıktan sonra geri dönüştürdüğü bir sistemi kurması, biyokütledeki potansiyeli süratle artırmamız için yaşamsal önemdedir. Bu potansiyel ayrıca enerji hatlarından uzak bölgelerde gelişen ve kendi enerjisini üreten yerleşim alanları, büyük hayvan çiftliklerinin kurulması için de çok önemli fayda sağlayacaktır. Bomboş duran tarım alanlarının kullanılması ve hayvancılığın geliştirilmesi açısından da büyük faydalar sağlayacaktır. Türkiye'de biyokütle potansiyelinin il ve ilçe bazında tespitini yapan BEPA'ya göre Türkiye'de yaklaşık 423 milyon adet büyükbaş, küçükbaş ve kanatlı hayvan bulunmaktadır. Bu hayvanların yıllık 194 milyon ton atığından üretilebilecek enerji miktarı ise 4.4 milyon ton eşdeğer petrol seviyesine ulaşmaktadır. Türkiye'deki 81 ilde ortaya çıkan belediyesel atık miktarı ise yaklaşık 32 milyon ton seviyesindedir. Bu atıklardan üretilebilecek enerji potansiyeli ise yılda 3.3 milyon TB ulaşmaktadır. Orman atıkları ve bitkisel atıklarda dikkate alındığında Türkiye'nin biyokütleden bugünkü kurulu kapasitesinin en az 10 kat daha fazla enerji üretebilmesi mümkündür. Tüm bu veriler Türkiye'nin diğer pek çok alanda olduğu gibi varlık içinde yokluk çektiğini ve potansiyelini en kısa sürede harekete geçirmesi gerektiğini ortaya koymaktadır. Bu duygu ve düşüncelerle etkin biyogaz üretimi ve güncel yenilikler çalıştayına katkı koyan tüm akademisyenlerimize, yabancı misafirlerimize en içten teşekkürlerimi iletiyor. Başarılı bir çalıştay olmasını diliyorum. Saygılarımla.
Well, Mr. Hüseyin Vatansever, you have just articulated my all feelings. You know, I cannot tell any more onto this one. Turkey is such an interesting and strategic country. It is so rich in natural resources. We have been living in a heaven, actually. But unfortunately, for some reason that I have been able to understand so many years, we are not able to exploit these natural resources in efficient way. We always look change our direction to Europe, to United States, and we search for the technologies which are already there. What with the important and crucial players just like yours, Mr. Denizsel, Mr. Ilchin, and you, and industry, we have such a strong and smart industrial people in Turkey. Why not we valorize it? And as I mentioned in my beginning of my speech, I mentioned that our potential is around 3,000 biogas plants, which is 9,000 in Germany, maybe over 18,000 in Europe. Why not we build another 3,000 in Turkey? But as Mr. Dennis said, already underlined that these reduced incentives will slow down these investments. It's obvious, but I see still potential that we can now press on the fertilizer side of the problem, and we can recover these loss from the fertilizer. And in Turkey, the overall estimation for these investments for 3,000 biogas plants is around 9 billion euro. Why not we just throw this away to Europe or United States? We can just build our own localized industry and so-called Andinization or nativization, whatever you call it. We have to have our local national technology, and this is obviously possible. Thank you for your contribution too. By that, I'd like to stop the opening speeches. It was really enjoyable and fruitful from my side, but of course, we have our other speakers, important distinguished speakers, and for our first session, Professor Dr. Yvette Ferret-Marty is anxious to initiate the first one. So I thank you all for your contributions, and I'm now transferring my turn to Professor Yvette Ferret-Marty, and I wish the other speakers good luck, and I wish a fruitful meeting. I see Professor Angladaki on the screen. I'd like to thank her. She deserves a really great and extra thanks for her great contribution. Welcome to our meeting, Mr. Angladaki.
Thank you. Thank you.
But Professor Marty, Ferret-Marty, stand is yours. Please go ahead, and it's your time now. Thank you.
Hello, good morning, everyone. Can you hear me well?
Yes, we hear you.
Okay. So good morning, everyone, and thank you for the invitation to chair this session. I'm very happy to be here with you today. So now we will start session 1 of this workshop, and the first speaker will be Irini Angladaki, who is professor at the Technical University of Denmark, and of course really well known in the field of anaerobic digestion. So please, the floor is yours.
Good morning to everybody, and I would start saying thanks for inviting me to the workshop, and especially to Professor Verdensi for this invitation. And I will now share my screen to start my presentation.
You can share your screen.
Yeah, okay. Can you see my screen?
Yes, we are seeing your presentation.
Okay, just a moment. The talk of my presentation is biogas status and future trends. And I will start showing you the agenda of my speech. First, I will talk a little about the biogas process, both historically, but also how it is nowadays. And I will concentrate
bugünlerde nasıl gidiyor buna bakın.
Management, because I know that Jean-Philippe Stein will cover other aspects, so we have coordinated a little. And then I will go to more future trends of applications of biogas, the biogas process. And I will start historically, because biogas is not something new. We have been known biogas for many, many years, for centuries. And in fact, Plinius and Van Helmont in the 17th century mentioned the biogas, and he was referring to mysterious flames appearing from swamps and subsurface locations. So they could see spontaneous fires from swamps, and this actually gave rise to rise of dragon myths. So they did not know where these flames came, so they thought that maybe dragons were the reason for these flames. Later on, Volta actually is recognized as discovering that methane was a product of anaerobic digestion, and Dalton established the chemical formula of methane. And of course, many of you know Buswell, who established for the first time the stoichiometry, and many of you probably have used this stoichiometry to calculate the biogas potential. So this is the knowledge of the process. And as to applications, these also have been for many, many years, for centuries. And actually they say already in the 10th century, it was described that in Assyria, they were using biogas to heat water baths, bath water, and also in Persia in 16th century. Also, the first sewer plant was built in Bombay in 1859. So we have a lot of stories of old applications using biogas. And in England, in Exeter, they were using street lighting in 1896. So as you can see, biogas is not new and has been used, and both as a process and also as application has been known for years. So nowadays, I think I have to remove this one. Yeah, there it is. Nowadays, biogas are modern facilities and they are used all over the world. And we have different types depending on the segment we apply the biogas. So you can see here in wastewater treatment plants, we have the anaerobic stabilization tanks, which are typically looking like that. In the agricultural sector, slurries, manures, other industrial waste that contain particulate matters, they are typically used, look like that. That means continuous stir tank reactors, big tanks with long retention times. While if we go to wastewaters, like industrial wastewaters, we have these high flow through reactors with immobilized biomass, such as, for instance, biofilm reactors, USB, EGSB. We have many different variations, where here we have much smaller, shorter retention times. And then if we have solid waste, then we have other challenges, that then we have some solid that has to be pumped, that has to be mixed up with the inoculum and so on. So we have different configurations again. So here we have a typical compogas laying down reactor. And we should not forget that we have millions and millions, again, rural simple biogas reactors all over the world. So in order to choose the type of reactor, you cannot really go from one segment to the other. So it does not make any sense to use thick solid waste or manures in an EGSB reactor, because that will be blocked and all the granules will be washed out. Or in a fully mixed with slurries, then you can, again, here you have to use a fully mixed reactor because of the particulate matter it contains. So that means in order to choose the reactor type, you have really to look into your influent material and to your feedstock to make the right decision for that. So there are extremely many different types in each segment. So they have standing up reactors here in the solid waste, the types. We have the Valorca type, the Dranko type, we have the Compogas. So there are many different and many companies offering different solutions. In this talk, I will concentrate on this type, the fully mixed agricultural type reactors, the fully mixed CSTR type reactors, biogas plants. And here you can see a typical biogas plant. These are all from Denmark, and they are consisting of 2 or 3 large reactor tanks. Here we have the pre-storage tanks. Here we have the after storage tanks. Here you have the gas collection order, and here you have the receiving tank, receiving building, where the trucks come in and unload their waste. And here you have the room where they have the gas motor and they produce electricity. So this is the typical last year. Food industrial waste, which it's taken there. You can see these plants have tracks. So this is a cubic meter track, which goes to the different farmers, takes the manures and brings it to the location here, comes into the receiving hall here, unloads through the hose underground the different substrates, which are pumped into the reactor later on. So this is more or less, and you can see here, actually, you close down the port, so all the odors are taken care of in order not to disturb the neighbors with odors and smells. So this is a typical arrangement. This is a bit old, actually, but more or less the same principle is used in other plants. So in terms of temperature, especially in Denmark, I think most of the biogas plants are operating at thermophilic temperatures. And this is for two reasons. One reason is, of course, that the reactions take place faster. The reaction rate is higher at higher temperatures, but that's not the reason they do thermophilic temperatures. The thermophilic temperatures, that means 50 to 55 degrees, the most of them, is because of the sanitation, because these co-digestion plants receive manures from many different farms and then deliver it back to other farms maybe. And in order not to spread out diseases, it's important that the effluents are sanitized, because the effluent from the biogas is an excellent biofertilizer, which is taken back to the agricultural soils. So the sanitation of it's extremely important. And you can see here heat exchangers, that means they minimize the heat losses, because Denmark, as you know, is a cold country, but however, the most of the biogas plants in Denmark are operating at the thermophilic temperatures. So you can really save a lot by having efficient heat exchanging. So the sanitation of manure is extremely important. Therefore, there is the effluence is extremely important. Therefore, we have various legislations and rules to how to bring it out before you bring it out for the co-digestion plants. And if it's mesophilic temperature you operate, that means 35 degrees, 37 degrees, then you need to pasteurize 1 hour, 70 degrees. However, if you run thermophilic temperatures in Denmark, then you can have a lower temperature for sanitation. And that means you need to have a minimum guaranteed retention time. For instance, keeping the effluent manure for a number of hours at a specific temperature before you pump it out.
Pompalamadan önce bekletmeniz gerekiyor.
Before they pump it out, it stays here in a holding tank for 8 hours at 53.5 degrees. If the plant operates at 50 degrees, a lower temperature, then this has to increase. So there are some rules how much time you need to have at a specific temperature in order to guarantee that your effluent does not contain any pathogens. So this is more or less what it's the current status in this type of reactors. And now I will come to something that is still lacking, and what we have not used up to now. We have not really taken grasp of the microbiology. There is the microbiology community management is still not utilized enough. And also another area that still needs to be updated, upgraded, is the reliable online monitoring for control. So up to now, the control of biogas reactors in this kind of plants is relying on the operator. So the operator decides which retention time, how fast, and what temperature and everything. So by having this online monitoring, then you can easily apply control algorithms to really optimize your process in the best way. I will not talk about this. I will just come into microbial ecology community management, because this is something that it will really take place and will really enhance the biogas in the future years. So this is the biogas process. We have a reactor here. The feedstock comes in, biogas is produced, here it's biogas, and the digestate is taken out, which is, as I said, an excellent fertilizer. So what we know now, it's basically the biochemical steps: hydrolysis, acidogenesis, methanogenesis. I will not mention them because I'm sure all of you are familiar with them. The process is a biological process. So the catalyst for this process is microorganisms. It's biological process mediated mainly by bacteria and archaea. We know that they are there, but we don't know who is there, what they are doing, who is working with whom. So we cannot manage the process microbially because we don't know exactly what is in there. So Then in the latest year, it was introduced the term microbial resource management. As we say human resource management, the same way we say microbial resource management. That means we need to know who our workers are in order to be able to manage them. And the term was introduced by Wilf Strater some years ago. And then in order to say, okay, we can go in and look to see whom we have there, what microorganisms we have there. However, we would have not been able to do it so far because the microorganisms fail to grow in the laboratory because many of them do not grow alone. They need each other, they live syntrophically, so they depend on other organisms for surviving. So we have not been able up to the latest years to identify all of them. However, now we have new methods and we have DNA that can help us in this. And all microorganisms, actually, all living organisms contain DNA, which encodes all the information needed to program the cell's activities. Reproduction, metabolism, specialized functions, everything is coded in their DNA. So all DNA found in an organism, microorganisms, is collectively called genome. So each microorganism have DNA, which is named, all of it is called genome. So the genome have genes. So you, in one organism, microorganism, you can have 3,000 to 6,000 genes. So if we have the genome, then we know exactly what this microorganism can do. We don't know if it does it, the microorganism, but we know that potentially they can do it. So we can take the DNA, extract it, and now we know everything about this microorganism. So if we have many actual microorganisms, then we have the metagenome, so many genomes. We can take a sample, extract the DNA, and then certainly we know everything we have in there. Maybe we don't have them physically, but we know what is there. And such methods actually have emerged to a new field, which is called metagenomics. So this is something that has happened the last 5 to 10 years, maybe more to 5 to 6 years, I would say, has taken off because of these sequencing methods have become cheaper. So as you can see here, this is actually the graph taken from yesterday. You can see how fast the metagenomics have developed. So this is the last 5, 6 years, which started from almost nothing. And nowadays we have almost 500 research papers actually reporting what is happening with these microorganisms. So now the last 5, 6 years, we will start knowing and being able actually to manipulate the microbiology in these cultures to have the most efficient process. So what we do is we take some from different reactors, lab scale, full scale, different processes, and we extract DNA. And with the DNA sequencing, we have the metagenomics. We can also extract RNA from the organisms, and then we have the metatranscriptomics. And we can also measure different parameters from the process. So with all this, with the metagenome, we can know who is there. Metatranscriptome, we can know what they are doing, what is activity, because here we know what they can do, but we don't know if they are doing it in a specific situation. But by looking the RNA, we know what actually they are doing at a specific case. And that means these tools will answer the questions, who is there, what they're doing, and how they are doing, how they are interacting. They will give us biological insights. So this is the approach. We have the reactors, different reactors. We take samples from here, extract the DNA. We make deep sequencing. We have millions of reads here. Then we put the pieces of DNA that we read from our sequencer together to larger scaffolds. And then we reconstruct the genomes of all the organisms. So here we have the metagenome. So these ones actually say exactly what we have in our samples. So now if I go to my old scheme that we did not really know, but only the biochemical process, now we can put names in here. You can see these red, green, and yellow points are actually microorganisms. And this shows that in the upper scale here, we have resilience. So that means many microorganisms can do the same thing. So when we go down here, we have specialists. So down here, we have only few microorganisms that can do only one thing. So it follows actually the funnel type, this anaerobic digestion. So here you are more flexible, so it's more possible that you will have problems down here if your specialist actually is attacked. Well, here you have resilience, so if one is gone, then the other can take over and so on. So it shows really how the stability of this process works. And we have also recorded by this metagenomics that there is a lot of synergy taking place. So both a synergy in respect to products and utilize as substrates, the hydrogen interspecies transfer, but also in axotrophy. That means some microorganisms produce amino acids which the other organisms are using, and in that way they save energy. So it's very complex, but we start shedding light into this complex process. As late as 2020, in collaboration with partners from all over the world, I would say, we have actually deciphered the biogas microbiome, and we found 1,635 microorganisms. This MAG means metagenome assembled genomes. That means we have the genomic composition, but we don't have physically the microorganisms. And you can see we have you can see the size of the genome is different here. You have from 1 mega base pair up to 10. So we have some very big microorganisms and some very small, and you can see a lot of the small actually are also present in the anaerobic digestion, and a lot of the small microorganisms are not capable of doing everything themselves. So that means they are syntrophic. So that means they live in collaboration with others.
Olduğunu tespit ediyoruz. Yani diğerleriyle işbirliği içerisinde çalışmalarına gerek var. Süreci konak hücrenin bazı özelliklerinden faydalanarak istila edebiliyorlar. Ama tek
I will not go so much into it, but we have a lot of knowledge, and I resemble that to as a treasure. Now we have a lot of, we can go in and extract extremely many different microorganisms that we know they have a lot of properties that we can use in optimizing our biogas process. So now I will go to utilizations of this knowledge, and we have ourselves and other colleagues have published a lot of papers of what you can use, what we call it bioaugmentation. And bioaugmentation is if you have a specific problem in your biogas reactor, by knowing the properties of the microorganisms, you can add the specific microorganism to circumvent a problem. For instance, if you have ammonia inhibition, there are you can find some ammonia tolerant microorganisms, and you can see that they are ammonia tolerant if they have osmo protectors, so they can somehow resist the ammonia inhibition. You can have solutions to long chain fatty acid inhibition and offer faster degradation. They accelerate the conversion of cellulose and hemicellulose. Now we have looked into them, we look at the way they work. Some of the cellulose and hemicellulose degraders, if you have a lot of grasses in your reactor, then you can add some of these microorganisms that we have found out how they work. They attach on the fibers. Some actually have other mechanisms. So according to what your problem is and what your process is, you can make somehow tailor-made solutions to your reactor by using the microbiology. Recovery from oxygen exposure of your reactors, overloading of the reactors. So there are really many papers. You can find the literature, the latest actually 2-3 years, I would say, so we have a huge advance in this kind of area where now we will be able to use the microbiology in different ways. Also, this is just to solve problems, but you can also develop specialized process indicators, which can tell you, okay, if you lack some basic, we have identified some necessary microorganisms that they are always there. So if your process lacks them, you can add them and recover the good balance of it. So that is the status, how it is now, and what we know up to now. So now, as my talk is also future trends, I will go in and mention you some future trends. And the future trends now is more integration of different processes. So integrating power to gas technologies. So you have, of course, I'm sure you have heard the power to X. This is extremely popular at the moment. It's really very trendy, power to X. So you use extra windmill electricity, extra photovoltaics electricity to somehow combine it with other processes. Gasification with biomethanation. Here again, we have the gasification pyrolysis platform, which is physicochemical. So there is also a lot of integration with biomethanation. So maybe you can take the syngas and ferment it to biomethane, so upgrade it afterwards and you put in the gas grid. So there is a lot of integration, bigger processes, larger units. I have here syngas fermentation. So now it's not only to use the biogas for heat and electricity. You go to other products like alcohols, acid elongation, etc. Because actually, as I will also come into later on, electricity production has really advanced very much in the windmill sector and the photovoltaics. They have become cheaper, so the electricity is provided ambient from these sources. Biogas upgrading is another trend that is very much in at the moment. And biogas upgrading is removing the carbon dioxide and having pure methane, which then you can use for other applications. Single cell proteins, this is now where we use the biogas to produce proteins, proteinous food. High value added products, platform chemicals, alcohols, bioplastics. So now we are moving actually really, and this is really taking place in very fast steps. So in my talk here, I will focus in these three topics and I will not go into this, although a lot is happening in here. And I will say this is not an extensive list. There are many other processes. So conversion of biogas to methanol. There are also pilot applications at the moment where you convert it catalytical to methanol, etc. Alcohols, etc. So there are many things going on in moving away from electricity production to other products. So I will take first the biogas upgrading to produce biomethane. So here we have our biogas reactor, we produce digestate and then 50 to 70% methane, the rest carbon dioxide. And the common application up to now was heat and electricity. However, if you remove the carbon dioxide and you go up to 95% methane, then you have certainly other possibilities here. You can use it in the cars, in vehicles, so as a transport gas, or you can put it in the natural gas grid and move it to other areas far away from your production, to cities, from rural areas to cities, for instance. So there are many different methods doing that. And this is a bit old, it's from 2016, but you can see already how many biogas plants have introduced upgrading. And I'm sure that this number is much higher now because a lot is happening in this area. So now we have buses running on biogas, cars running on biogas. They have got permission, at least in Denmark, I know, they have got permission to put the upgraded biogas in the natural gas grid. So then the natural gas becomes actually then CO2 neutral compared to the fossil natural gas. So this is not just an application that will happen, it's already there, the biogas upgrading. So there are many commercial methods, water scrubbers, pressure swing absorption, amine membranes, etc. But the method that we have developed is a biological method where you capture hydrogen, carbon dioxide with hydrogen through this reaction. So you capture your carbon dioxide from biogas with hydrogen, injecting hydrogen, and then the hydrogenotrophic methanogens will convert that easily to methane. So in this case, we need hydrogen. And where can we find the hydrogen? We can find it actually from the periods where we produce too much electricity, excess electricity from our windmills. We can electrolyze water to hydrogen and oxygen, And the hydrogen can be put in a biogas reactor where it can capture the carbon dioxide, leaving the methane pure. But at the same time, as you notice here in red, we get more biogas. So this is actually, we call the biological upgrading method. We call it second generation upgrading. And this is very popular because we have also capturing of the CO2. All the other methods, the commercial ones at the moment, they are not capturing the CO2, they're throwing it out. So this is a second generation where we capture the CO2 capturing is extremely important, and especially Copenhagen municipality wants to remove carbon dioxide because they have the goal to become a CO2 neutral city by year 2025. So they put a lot of attention in any kind of method which will capture the CO2. So there are different ways you can do that. The biological upgrading, we have here the electrolyzer, produce hydrogen. We can either put it directly in a biogas reactor, where here it will capture the CO2 and will have pure methane out. This is called the in situ process. This sounds as the most direct and easy and cheap. However, here you actually influence and intervene with the pH of the reactor, because by capturing the endogenous CO2, you will risk to increase the pH. So there you would need to make in co-digestion with acidic waste somehow to keep the pH buffered at a level which is not detrimental for your microorganisms. Alternatively, you can have ex situ. That means you put your hydrogen together with your biogas in a separate reactor. And here you don't have the problem with pH because this is not endogenous CO2 produced. So this is the ex situ process. You have also the possibility to make the hybrid process, where you make the upgrading directly in the reactor here up to maybe 80–85%. And then you have a polishing step where you can go up to 95% or how much is required for you, 98%. Where here the reactor, of course, is smaller because you have less carbon dioxide to remove. So this was already published in 2018. So another issue that it might be important in this biological upgrading method is if the wind is not blowing for a period, then you will not have excess hydrogen, excess electricity, and maybe you will need to stop your upgrading, how your reactor will process. So here we have stopped the provision of hydrogen for 1 week, restarted. So we stopped it here, we restarted, the process revived very fast again. Again, we stopped it here, restarted at the green line, stopped again, and then the third time. Then we had the longer stop here, 2 weeks, and again, it started again to 95%, although it took a little longer time. What I mean by this is that although you don't provide the hydrogen continuously, your process does not need to entirely restart. It's there, the microorganisms are still alive, waiting for the new shot of hydrogen. So another challenge in this biogas upgrade is the gas liquid transfer. You provide hydrogen, which is liquid, and this has to reach your microorganisms which are here. So it has to pass through the gas liquid transfer interface here, from the gas to liquid. From the liquid has to travel to the microorganism. You have another barrier here, the liquid to cell barrier, and then has to go into the cell to react. So that means here you have all this way to go through. And therefore, we have tested different membranes, different pore sizes, different materials, immobilizing materials. I will not go into the details. We have made many experiments. And the problem is, of course, if you have long retention times, it wouldn't be any problem. But the thing is that we need to go to very short gas retention times, as you need to have huge reactors. So this is the gas retention time. We start from 4 hours, 2 hours, 1 hour, half hour, and 0.25 hours. So apparently around below 1 hour gas retention time of your gas, then it starts being prone to use it, all of your hydrogen. So it is possible to go down to these gas retention times. So then we have actually constructed, this is the schematic representation of a pilot reactor, and we found out that the best way to do it is a trickling bed reactor where you feed your gases from down, and you shade here, you wash it with digestate, and the role of digestate, it's just very slow, maybe twice a day or something, washing it with digestate, providing the nutrients for the microorganisms, because these microorganisms would need nitrogen, phosphorus, etc. to live, and then in order to consume the gas. So the gas comes up while you wash it slowly with the digestate. So here you can see the pilot reactor itself. It's not, and you can see here the gas is coming from down, and here you can see the pumps putting the digestate through here down to wash the bed of the microorganisms, which is immobilized in some materials. So this is has been tested. The first attempt was tested at Avedura wastewater treatment plant. It was for the Vaga project, and you can see the setup. And then later on, it was tested in Levi Biogas plant. And in both cases, we could reach, in some instances, methane content of almost 99%. So the last phase now I'm with a consortium coordinated by a company called Nature Energy, and we have partners from university and another company, Clean Gas. This is Clean Gases reactors. It's a trickling bed reactor, and this is at the stage where they want to upscale it. So this is seems that the technologies will maybe, I don't know, soon will come to the market. This is another similar concept, which is called electrocare, and it's a smaller reactor here, thinner, I can say. And the concept operates with pure culture. Our concept is actually mixed culture, because we let the hydrogenotrophic methanogens establish by themselves, because they are everywhere. In any biogas reactor you have hydrogenotrophic methanogens, meaning you have methanogens that can take hydrogen and CO2 and convert it to methane. So that's gives more robust process. So the challenges for biological upgrading, as I said, gas liquid transfer, configuration, the filling material is important, pressure maybe needs to be slight overpressure to be applied. In microbiology, you might sometimes have competition between homocetogens and methanogens. That means instead of doing methane, they might make acetate instead. And also that the process, exothermic process, which might require cooling, although I have not seen it at the scale we run, we have not seen problem with too much temperature. But this has to be considered depending on the surface volume ratio you have. So that was the first application. So this is the future applications I was mentioned. The first was to make biogas, biomethane, and the second is single cell protein. So now we'll take our methane and make proteins. And there is a need for protein, 100 edible protein per person per day. And agricultural soil is limited, so we need alternative sources for proteins. And then the question is, can we use microbial protein? And yes, indeed we can, because we would need a lot more protein in the future. The population in the world increases, so we are going to need a lot of more proteins that maybe the agricultural soils cannot provide. The food supply actually might be an intense problem in the future for the future populations. So here, when we say microbial protein, cells are like, for instance, meat, they contain protein. So single cell protein refers to single cell microorganism with protein content. They can be mixed culture of algae, yeast, fungi, and bacteria. And here you can see the different compositions. And as you can see in red here, we have the bacteria. They can have 50 to 70% protein content in their cells. So they are really very rich source of proteins. They have low fat content, a little ash, and this is a bit negative. They have nucleic acids are not wished as food. Therefore, if we need to use them as human food, we need to. It's EU approved to use for some animals. So the process is not new. It has been already discovered and used in the 70s. We have a Danish company called Unibio. This is the Unibio installations here. another company Calista, etc. And this is the Unibio concept, but all of them, they run on natural gas. That means they run on fossil fuels. We want now to convert the process and make it to operate on biogas instead of natural gas. So we cultivate some microorganisms which called methane oxidizing bacteria. That means they use methane with oxygen, and the cell mass is the feed. So it's proteinous cell mass. So you can see here the Unibio concept. You need to mix oxygen with the methane. So they have a special arrangement called the loop reactor. And when we go here, they take the microorganisms, they dry them, spray dry them, heat, and then down here they get the pure food for the animals. So here you can see the installations of the Unibio. Unibio is actually a partner in our projects. And here it's their product. And if you read down here, it says that it is EU approved. They make this nice food for animals. So the waste and wastewaters are produced in large amounts in cities. And therefore our vision is to use these resources to produce food, which actually will return both the carbon, but also the nutrients back to the cities. So we take all this wastewaters, we make biogas here, you see the anaerobic digester, and then the biogas is upgraded biogas, and at the same time, actually, we extract the nutrients, here the nitrogen. So we feed then the nitrogen, the nutrients from the digestate, together with the methane from the biogas, we feed it to make single cell protein, and this is taken back to the cities. So this is a total recycling of our city waste. And here a Copenhagen municipality is involved, the Amager Resources Center, so they are involved to diminish all the load, both in respect to carbon, but also to nutrients, and recycle it back to the cities. I mean, to make a total bioeconomy approach. So this project has been funded, and we are about to finish with that. So the idea is, here you can see it schematically, take all the waste from the city, make a biogas. Biogas is upgraded here, put it, and then here we retrieve the nitrogen through an electrochemical system, and we feed to make this nice protein food and feed to the chicken and then back. So this means that in the future, it's not our customer is not only electricity, but we have many possibilities, and this will give a much better price to the farmers that they produce the biogas. So we investigated here because we have some issues here. Is the carbon dioxide the problem? Are the nutrients enough? Is the hydrogen sulfides in the biogas might cause a problem? And therefore we made some batch experiments here. We tested both with the synthetic media and the natural gas, and then we with biogas, with digestate, and with a digestate where we have retrieved the ammonium. And in all cases, we could make actually very nice proteins. You can see the composition is more or less the same. And we also identified the microorganisms produced. And we have two main types. The blue is the methylomonas, and the yellow is the methylophilus. So these two types always are the dominant, and they live syntrophically. This one actually can oxidize the methane to methanol, while this one takes up the methanol and use it to produce single cell protein. While this one is inhibited by methanol, this helps it to reduce the methanol content. So that means actually they help each other, and they are always appearing in the composition, these two microorganisms. Then we looked into, is there something in the biogas? Because this is with the natural gas, this is the biogas, it gives lower yield. What it is that makes the yield lower with the biogas compared to the natural gas? And we suspected the hydrogen sulfide. We investigated. So the hydrogen sulfide actually has to be removed, because you can see here already from over 1000 ppm, it's really inhibiting the growth of these methylotrophic microorganisms. So hydrogen sulfide can be a problem. The other thing is, can we use the digestate alone as to supply our nutrients? Here it's with the synthetic medium. This is with the digestate, and you can see with the digestate it is lower. So we looked at what lacks in the digestate. So we measured throughout several seasons. This is a synthetic medium. These are different sampling times of this biowaste. And we can see especially actually, copper. This is in the medium, this is in our composition in the waste. So we might lack copper because the enzymes promoting this reaction, they are catalyzed by copper. So they need copper to action. So we would need to add additional copper in our reaction, and maybe also iron is deficient. But now we know a lot. We know that we can do it. We just need to optimize in some places. You can see here the copper. This is the synthetic medium, and this is the real concentration in the digestate. So that means we can use both the methane and also the nutrients from the digestate and produce proteins. Here it's actually where we try to, I have to hurry a little up here, I can see. We try to use the electrochemical system to extract the ammonia from the system. I will go to here. We made the 2 steps process where we managed to, in the cathode, to extract ammonia of 60%, over 60%. And this is actually not optimized. I'm sure we can go maybe to 60 to 80% recovery of the ammonia of the digestate, which we can feed to our microorganisms. So that was the second application. We made it also in a continuous scale. You can see here is the growth, and these microorganisms, they have this typical pink color, this culture. So they are pink, these methylotrophic microorganisms. You can see here the amino acid composition of these proteins were produced. They contain all the essential amino acids. So it's really a very good source of protein. So we managed to convert waste to feed, actually. So the next example I will mention, I have only a few minutes left. It's this biosuccinic acid. You can see this is biosuccinic acid, white powder. It's platform chemical. We have thousands of different applications all over the world. You can see here how many different applications you can use it, to cosmetics, to pharmaceuticals, to food application, industrial applications, etc. And has a price which is around, depending on the purity, over €3 per kilogram of this product. So that means, again, you can go in higher product. And usually it's produced by petrochemical route. We can make it in by fermentation mode. And in this fermentation mode here, you can see it's actually using also CO2. So we can use our biogas to capture the CO2 here together with the waste and produce the succinic acid. So this is at ambient temperature. This is a petrochemical, very high temperatures. We go low temperatures. They emit carbon dioxide. We absorb carbon dioxide. So it's a very good process. And for this carbon dioxide, we can use the carbon dioxide from our biogas. So this is a process where we can feed biogas. The microorganism can take up the, we can take up the biogas here and produce succinic acid, pure succinic acid. And because we have removed the carbon dioxide, we have also upgrading of the process. So we have a EU project at the moment, so we're about to upscale it, and the coordinator is Norvento from Spain. It's a fast track innovation project. We'll finish 22, probably we'll get extension because we started during the COVID-19 crisis here. And at the moment we are about to build the pilot scale reactor. Here it's the container, the reactor has been purchased, and we are very soon to have the installations ready in the production. So here are the countries participating. As I said, Norvento, IVEM, DTU is the university, and this is another university. And the manager is the IVEM from Spain. So here it's the process. We have promised actually to make pure methane with this process, and the pure for transportation, and then also pure biomethane 95%, and the biosuccinic acid 80% purity. No, 80% yield, and the purity should be 95% of the biosuccinic acid. So we work both upstream and downstream in this project. So the conclusions to finalize is that the future, it's biogas upgrading. We can do it biological with hydrogen eutrophic methanogens. That is successful, and it's very close to commercial level. We can also produce biosuccinic acid. Here we are in collaboration with a company which is actually ready to take that to commercial level, although it's still the pilot has not been implemented on biogas. The biosuccinic acid is another option. And I want to say that is just some examples, because there are a huge number of attempts. You can make a lactic acid, you can make acetate as a product, you can make polyalkanoates, hydroalkanoates, etc. So there are many, many different products, and they are working all over the world, and many have been upscaled, some are still in the lab scale. And that's more or less my input to this discussion. And thank you very much for your attention.
Thank you, Rini, for this very nice and inspiring presentation. And the chat, there are a few questions, but as we are a bit late, I propose to do these questions in the coffee break. So then I think we should now move on to the next speaker, who is Jean-Philippe Steyer. He is a senior research director at INRA in the Laboratory of Environmental Biotechnology. So good morning, Jean-Philippe, and the floor is yours.
Thank you very much, Yvette. Do you hear me? Yeah?
Yes, we can hear you well.
So first would like to thank the organizers for inviting me. It's always a pleasure and an honor to present our work. And I wish we would be all together in Turkey. I had the chance to visit Turkey, and I really enjoy your country, and I met amazing people there. So maybe next time. And so today I want to present to you some innovation again to improve the biogas economy. So I work in a lab in the south of France. And just a few words about the interest of anaerobic digestion. We have to remember that some years ago, anaerobic digestion was just a wastewater treatment facility. And now it's more about energy production and digestate also as fertilizers. But I will show you that as Irini presented to you just before, that there are many, many other options. So the first thing is wastewater treatment, urban wastewater treatment. And if you look at the wastewater treatment, you have aerobic line, and you have the digester at the end that will digest sludge. And in these conditions, you remove pollution from the wastewater, and you have a clean water at the end, but you consume energy. And so over the years, about like 5 years ago, this has changed, and from wastewater treatment plants to water resource recovery facilities, which is a different options. And what you see here, it's just an example, as if you make the maximum of sludge and the maximum biodegradable sludge, you can put the anaerobic digestion up front, and you will produce energy. And so the first aspect is energy. But there are other anaerobic digestion, reduction of greenhouse gas emissions, but something which was also mentioned before in the introduction speech, it's creation of local jobs and income diversification for the agricultural sector, which is also important. And one aspect that will change a lot the way for energy, the way that anaerobic digestion can play a role is the fact that now, and I'm sorry it's in French, but now the gas grid and the electricity grid, they can talk to each other, which is a big change because it's difficult to store energy in terms of electricity, but it's easy to store natural gas or renewable gas. And in France, for example, just the pipe, the volumes of the pipes, it's 4 months of energy consumption in France. So as Irini showed, you can transfer surplus of energy, surplus of electricity with either electrolysis or methanation or anaerobic digestion and go from electricity to natural gas. And this has changed a lot the picture of the way anaerobic digestion can be involved. I will not go into too many details here because it was shown before, but the surplus of energy, you can produce, for example, hydrogen that will be converted to more methane. And as it was shown, limitation of gas transfer is a big issue. So you have to solve these things. But also anaerobic digestion microbes, they can produce hydrogen. Here you have the metabolic network of anaerobic digestion. And what you have here is hydrogen. And if you apply some specific conditions, you will lower the activity of the methanogens and even remove them if you want, and you will produce hydrogen. And so we work a lot on these things. And what you can see, it's you have a two-step process where first you have the dark fermentation process where you produce hydrogen and the remaining waste is treated by anaerobic digestion. And this works well. The main aspect is you have to have as much as sugar as possible in your filling tank because the quantity of hydrogen you will produce is related to the sugar content of your waste. And now we have in the process of making a demonstration plant where from organic residues, you will combine these two steps and you will produce either methane or either hydrogen for local mobility solutions like vehicles or tractors. And you will also produce methane. So is there any remaining challenges? Well, many of them are remaining. And for example, if you go to a refinery, an oil refinery, what you have is you extract oil. You have a facility where you refine the oil, and you will produce different source of energy and also different materials like plastic or PVC. And if you do this, you can do the same with a plant. So this is called a refinery where you collect vegetables or plants and you refine them and you can produce many things. It's also energy like bioethanol, but it's also many different things. And an environmental biorefinery is the same principle, but you use waste. And from this waste, you can have many outputs which are not only energy. And this is, as Professor Angelidaki very nicely showed, this opens very different and exciting valorization of the waste. The constraints, however, compared to oil and plant is if you have to deal with multiple waste, multiple inputs, which will vary over seasons, over days, over weeks, and this change a lot. And so you have to play here and you have to ensure sanitary and environmental constraints. And this is pathogens, this is micropollutants, this is different aspects that you have to deal with. And in order to produce energy, molecular platforms, fertilizers, and of course, clean water. And you have to think circularly, and the single cell protein scheme that Professor Angelidaki just showed is a very nice example, going from either solid, liquid, gas waste to recycle to the city or to the agriculture. And this is where anaerobic digestion can play a role. one aspect is if you, roughly, if you add 100% here, 100 kilo, you will have 50% in terms of methane and 50% of digestate. And you cannot maximize both. So you have to make compromise. If you either you focus on biogas or either you focus on organic fertilizers. So this is important to take into account. Just a few words about digestate. And a nice aspect is if you go from the needs of soil and plants, you can design the way you operate digesters in order to optimize the need and to bring what is needed for the agrosystem, soil and plant. So you can engineer what you do to maximize the content of the feedback for agriculture. And I will not talk too much about this today, but you have to keep this in mind. And this is also important for farmers. To go further and go back to anaerobic digestion, to go further with targeted services, we discussed methane, we discussed hydrogen, but as it was shown also before, you have many molecules that can be produced, either acetates, organic acids, and different alcohols. And you can valorize this also as much as possible. One example in complement to the presentation just before is you can use microalgae here. You have the waste. Here you have the dark fermentation where you basically produce hydrogen, CO2, and some volatile fatty acids like acetate and butyrate, and you can feed it to microalgae where in mixotrophic conditions you can produce either biomass for protein, you can also produce pigments, different lipids, biofuels, but also for cosmetics and skin, and also carbohydrates like starch that can be used furthermore. So this is another valorization possible that you can do with your digestate. Digesting meaning the output of dark fermentation in this case. You can go even further. It was briefly mentioned before, bioelectrochemical systems. If in a reactor you put an anode and a cathode, and you put in the middle, you put a membrane, well, you have some specific microorganisms that can transfer electrons. And if you do this, well, you can produce electricity as a microbial fuel cell. But the most important is not this, because you will always be limited by the power you can produce per volume of reactor. You have here an example. So you have a reactor, an anode, a cathode, and a membrane here. So what I want to say is if you apply a difference of potential between the anode and cathode, so you apply a little bit of energy here, you will bring electrons to the microorganism, and you will have a way to react and to play with the function of the microorganisms. So this is an important aspect. The second aspect is here you have the part of the reactor where you have the waste, and here you have you can have a pure culture, and this is a way to make wastewater ecosystems connected to biotechnology, industrial biotechnology, and you can make different products. And this is for me important. There's very nice studies where this has been studied. So you have an hydrolysis step here, and you will either go to the biogas plant or either go to microbial electrosystems here, where you will also take advantage of the electricity and the CO2, and you will produce targeted molecules. And at the same time, you will produce also electricity and digestate. So, of course, as I said at the beginning, organic waste are changing and fluctuating over time. So what you can do is you can imagine a network of these bioelectrochemical systems where according to what you have at the input and in terms of composition and variation, you can send a part of your waste to a network of bioelectrochemical systems where you will have a real nice environmental biorefinery to refine residues. And this is a project that has been running by one of my colleagues, the Biogas project. Also, as it was highlighted before, methane is more than energy, and methane can be refined itself and to produce different chemicals. This is an example, and again, I will not go into details because it was just presented before, but this has to be kept in mind. One main advantage of this is if you refine biogas and you use different chemicals, you get rid of the sanitary issues because you will have a pure molecule that you can use, and in terms of legislation, this can change a lot the problem. And just to conclude, to be brief, any remaining innovation? Well, many, as Professor Angelidaki presented before, just want to highlight something. This is a study we did some years ago, and the idea was, okay, here in terms of axis, you have the technology of the digester, and here you have the organic loading rate. And in terms of, according to the waste composition and the technology, in terms of kilogram of COD per cubic meter and per day, the best you can achieve, it's 70 kilogram COD per cubic meter and per day, with a very biodegradable substrate and mostly liquid substrate. But when you look at nature, the story is different. The story and the cow, for example, is 220 kilograms COD per cubic meter and per day, and the termite is 400. So what we investigated is, well, if we keep on looking at digesters the way we look as engineer, maybe we will achieve 80, 100, but how come we could achieve 400? And maybe nature over the million of years of evolution has found solutions. So we investigated the digestive tract of different animals, and we investigated 190 digestive tract different animals. And the question, well, what is the main influencing parameter? Is it the feeding, the waste, the composition of the food? Is it the way it is operated, the digestive tract, or is it the way it is designed in terms of volume? And when you look in our digestive tract, according to where you are in the tract, either you have a batch reactor, either you have a CSTR reactor, either you have CSTRs in series, or either you have a plug flow reactor. And if you combine these 4 different configurations with pretreatment meaning either pH pretreatment or enzymatic pretreatment, and the enzyme can come from the host, or the enzyme can go from the microbial ecosystem directly. So you end up with 24 different configurations. And if you look in nature, out of this 24 configuration, well, nature has selected 8, which means if you want to apply for optimizing the product recovery from a waste, and if you want to have an acidic pH pretreatment combined with enzyme coming from the ecosystems, well, maybe it's not a good issue because nature has never selected this configuration, and you should have from enzyme coming from the host, or exogenic input of enzyme, and you should avoid plug flow. That's one output of this study. The second output is, if you look at the food, the animals, they can be carnivorous, they can be omnivorous or herbivorous. So it can go from readily biodegradable substrate to slowly biodegradable substrate. And if you look about the design of the digestive tract, well, in fact, you have big changes between herbivorous and carnivorous. For example, you have, in terms of volume, you have big volume at the input and very low at the end. Whereas most of the volume of the digestive tract of carnivorous, it's at the end of the treatment of the tract. And if you look even more in the herbivorous configuration, here you have 4 examples. The cow is this configuration, the llama is this one. The kangaroo is an herbivorous, but it is the only one which does not produce any methane. And you can see the configuration of its digestive tract is quite different from cow and llama. At the other hand, you have oasin. Oasin, it's a bird which is not herbivorous, but it's a bird which has a very close configuration to the herbivorous configuration. And the amazing output is it's the only bird which produces methane. So the way the digestive tract are designed is influencing the output in terms of methane. And maybe this can have an input also for digester configuration. So we have a lot to look into nature to find optimization and new routes for anaerobic digestion. Last but not the least, we analyzed the microbial composition of the feces of 190 different animals. And here you have the microbial diversity of the ecosystems. Here you have the weight of the animal. What you can see here, you have the big animals, and this is something we didn't expect. Big animals like elephants, giraffe, and here you have the birds. Here you have humans in different size. And what you can see is the bigger the animal, the more diverse is the ecosystem. And this also can have an influence on the way to operate digesters. And with this, I wanted to be short because I knew that Rena would be a little bit long. I conclude my presentation and I would be very happy to answer any questions. Thank you very much.
Thank you very much, Jean-Philippe, for sticking to the time. Yeah, because we are a bit late. So these have been 2 really nice presentations. So now we have some questions from the audience. So the first one is to Professor Irini. So can microbial communities retain their distribution and activity during many batches of operation? How can you do to maintain their stability? Could you hear me well?
This is a very good question, and it's a very difficult question to answer. I would say that we would assume if we have constant conditions and constant feed, that the microbial community will not change, it will be stable. However, we have been operating an upgrading reactor for several years, and then we saw a gradual drift of the microbial community. So there are still many things that we don't know exactly how they are, because the feed and everything were constant, temperature constant, everything constant, and we saw a drift of the microbial community. So we have investigated that there are some advantages of some microorganisms, and then maybe in respect to the growth rates, it takes time but I mean, it took like 2 years to change the community. So if I would answer that, I would expect that this should be dependent, the stability would depend on the stability of the operations and the feeding composition. So that would be my answer to that. And I would say to Jean-Philippe, I'm sorry that I took from your time. I prepared the presentation yesterday and I didn't know actually that how long time it would take. Yeah.
It's fine. They have both been really interesting.
Can I add something?
Yes, of course.
And of course, then no problem. No, just one thing about the way to play with ecosystem, microbial ecosystems. There's something we forget sometimes is we can change the outputs we get from an ecosystems by augmentation or playing with the ecosystems. But there's one thing that is we not mention very often is, for example, in the bioelectrochemical system, if you add electrodes or if you add some specific microbes, one main output for me in terms of application afterwards, it stabilizes a lot the results. The variability of the results, if you make triplicates with normal ecosystems, well, you can have a standard deviation like this. But what you can get also, it's the same output, but standard deviation will be like this, which will impact application further. And because the downstream process will be facilitated. And so we have to also think about this in terms of stability of the results we obtain.
Okay. We have some more questions. So again, for Irini, how much can the output increase with proper understanding and managing the biogas process through microbial resource management? So I see.
Yeah, this is actually can be really very much. We have made the experiments with ammonia inhibited process where some feedstock like proteinous waste which contain ammonia or pig manure or some this kind of waste have a lot of ammonia. So we operate actually stably. We have a stable output. However, this output is lower than what it should have been because it's at the state which is called inhibited steady state. So you get every day the same amount. It's stable, but it is low. So we have tried to make a augmentation of this inhibited steady state process, and we got an increase of 40% of the biogas output. So it can really be a lot. So it was, and the biogas operator had no idea that they were actually working at so low outputs because they said, but I get biogas every day, it's fine, there's no problem. But apparently then there was a problem and we could fix that with augmentation.
Very good. Another one. What about using the use of recombinant bioenergy plants? So that means plant cell wall genetically modified for future studies. Do you think this could be used more widely in the future? So using recombinant genetically modified organisms.
You mean microorganisms, gen-modified microorganisms?
Yes.
I think technologically, I wouldn't see any problem. We could use them if they are stable and robust. But I think this is a matter of legislation, which, plants, I can see, yeah. Yeah, I mean, the answer is the same. I think this would be depending on the legislation. But I think as long as they are approved as food and plants, then it will also be approved to put in a biogas reactor.
And then we have, how do you plan to commercialize the new success project? For example, how can oil refinery be involved in your exploitation model?
Yeah, the no success project we run at the time is coordinated by Spain, and there are two different companies, and the one is a renewable energy company and the other is build biogas plants. So they intend actually to develop the technology in vicinity of biogas. So they have access to biogas and then they have access to waste, and there they will actually commercialize that. So that's the idea of the project.
Okay. Then we have one question for Jean-Philippe. Bioelectrolysis is a novel approach. I wonder about the energy and water consumption to prepare solid waste for the electrochemistry. Have you done any optimization?
If, for example, if you can produce with bioelectrochemical systems, you can also produce hydrogen, for example, as a cathode. If you compare this with electrolysis of water, you produce the same amount of hydrogen, but with 10 times less energy required to produce hydrogen, for example. This is just an example that microbial microbes can have a very positive input in terms of energy consumption. And there are a few studies on, for example, LCA of bioelectrochemical system, and they show also great input, great advantage of doing this. So this is available in the literature. Yeah.
Okay, thank you. And then one more question for Irini. I think it refers to the upgrading in slide 25. It says hydrogen is put in a different bioreactor. Is there any bacteria in this reactor?
25, you say, just a moment.
Slide 25. Hydrogen is put in a different reactor.
Yes, yes. The answer is clearly yes. The process, the biological upgrading, is performed by hydrogenotrophic methanogens. So they need to be there. So in all reactors where we want the process to take place, the conversion of CO2 with hydrogen to methane, there are microorganisms. So maybe in the photo they look like empty, these trickling bed reactors, but they are filled with filling material. On the filling material, they are attached microorganisms. So there is a thick black biofilm built on this filling material.
Okay. Are there any other questions from the audience for Jean-Philippe and Irini? If not, I think we can all go do a short coffee break and then come back at 10:05 to continue with this session.
Okay.
Okay, see you in 7 minutes.
Hi, Michael.
Hello, Nuri.
Yeah, before we start, I just wanted to say a short hi to you, and I'll transfer to Professor Yvette now. Thank you for your contribution. Okay,
yeah.
See you later then.
See you later.
So I just need 10 minutes after your presentation, okay? Thank you.
So yeah, I'll take about 20 minutes.
Okay, good. Thank you.
Yeah.
Thank you. Bye-bye.
Yeah.
Bye-bye.
So hello, everyone. I hope you all could have a quick coffee or tea in this brief pause. So now we'll move on to our next speakers. In this case, we have two. Michael Kodner, who is a senior expert consultant of the International Biogas and Bioenergy Competence Center, also known as IBBK, and also Professor Nuri Asbar, who is, we all know him by now, who's full professor at the Department of Bioengineering at Ege University in Turkey. So the floor is yours.
Yes, let's try if I can share the screen. Okay, can everybody see the screen? The share screen?
Yes.
Okay. Yes, thank you. Good morning, everybody from Germany, and thank you very much for the invitation. It's a great honor to speak in such a large conference. I saw that you over 200 people. That's quite impressive. Congratulations, Nuri, to that. I wish we could all join together in Turkey and enjoy the spring, but now we are at home. It's also some advantage. But yeah, let's hope we can meet soon again. So let's try and do this presentation together with Nuri. I will focus on the development and the circumstances in Germany and Europe, and then I speak a little bit about practical innovations. We as IBBK also do a large conference together with the University of Hohenheim with Hans Oechsner. We were also due to do it this year, but we will also do it online in September now, because I don't think it's possible to travel this year. So we will also do it in September. So my introduction, a little bit about IBBK. We do know-how transfer, we do workshops, conferences, also do technical support. We have a lot of contacts with experts in planning and design, and do networking. The content of my speech is the overview of biogas and biomethane production in Europe, feasible technology options for dry and wet residues and the innovations, some in economics and some case studies. So yes, what is the state of the art at the moment? And I'm doing a lot of I'm asked about that quite a lot lately, especially from investors. So there seems to be a lot of interest to invest in biogas projects, and especially in this renewable energy sector, because we can not only produce electricity, we also produce gas, we also produce renewable gas. And if we look at it, at the total installed capacity, electrical capacity, it's over 11 gigawatt in Europe. And we can see that Germany has about 2/3 of that share. And it's over 11,000 biogas plants altogether, including landfill gas and sewage treatment. The German Biogas Association always only counts the agricultural and the waste plants. They're about 9,500, 9,300. This is all included here. So is it included with all the other countries here? The second at the moment is Italy, then the UK, and then it's France. But these are statistics from 2019. Now we are in 2021. And from single countries, I know that there are many more biogas plants, especially it's growing quite quickly in Italy, in UK, and in France. Also in Denmark, it's also growing quite quickly, especially in the biomethane sector. So there are still favorable conditions out there which make this industry grow. But we have to fight, and I will come to that later. Also, when we look at biomethane, in Germany, we have, out of the 11,000, we have 232 biomethane upgrading plant. And those 232, they consist of 10% of the biogas production in total. So these plants are quite big, they're quite large plants, they're quite large production plants, and 90% is actually used for electricity production. But in Germany, also the biomethane doesn't go only into the grid, it's taken out again and used in cogeneration plants largely. So this is what the status is in Germany. In France, these figures from 2019, they are now overrun already. We have over 150 plants here, and also in the UK, there are also 130, 140 plants of biomethane upgrading. So this is growing very quickly. Also, Ireland, there was the year before there was nothing. Now there are 11 plants. So there's a lot of growth in the biomethane sector, and a lot of these countries, they are count on the green gas from biogas. And the green gases, they are very much on the political agenda, especially now when we talk about green hydrogen from excess electricity. What we also observe, a strong growth in the sector for bio-CNG and bio-LNG and heating use. So in Germany, away from only using it in the CHP, but going into the transport, into the short transport, so it's like cars, and LNG for trucks, buses, and for ships. So this is now, and then when the big players are coming in, it's Shell, it's Total, it's the oil and gas companies who invest in bio-LNG projects and even buy them. Especially in northwestern Germany, biomethane from manure is on the high agenda because we have a manure surplus area here where the farmers don't know where to put their manure because of too many nutrients. So manure collection stations are built, and they are producing biogas and biomethane, and they're injecting it into the grid. And near Cologne, Shell is building a big LNG station, a converting station. So also bio-LNG can be offered in more and more filling stations. Last year we had 10 LNG stations. This year already, or in 2020, we already have 37 LNG filling stations. So a lot of things are going on there. Also in Italy, in 2018, there were no biomethane stations. Now there are 12 in 2019, and there are even more to be built in the future. The same is France. France has 140 as of November 2020, and there are 1,100 new projects in the pipeline. So you can see it's quite a big uptake, and more biogas plants are converting to biomethane. So here we have the scenario in Germany, like regions with very many biogas plants and some regions with less. In the regions with many biogas plants, we also can think of connecting them, interconnect them with raw gas pipelines, and leading the raw gas to a central upgrading station to be upgraded and injected into the grid. That the first few projects have been realized already, and I think there will be more to come. What is a feedstock? Still, we have a lot of energy crops here. About 50% is energy crops, and then it's manure and residues from agriculture and industry and communes. But due to politics, these energy crops are due to change, especially high-yielding energy crops, maize silage. This is not very liked by the politicians. So in Germany, there's a cap of now 40% of corn silage. In France, there's a cap of 15%. In Italy, it's, I think, 20%. So these plants, these high-yielding plants, or the cash crops, they are supposed to go into food production. But the politicians need to be taught that if they are taken out of production, it doesn't mean that we have lower CO2 emissions. Corn is still grown then and used for food production. So, or overproduction. So there still something needs to be readjusted. And if we want to have flowering plants, or if we want to have plants which catch the CO2 in the soil, we need to still change the politics and tell them what to do. Yeah, in the past, there was a very strong growth of biogas plants in Germany, almost 7,000 plants in 10 years from 2004 to 2014. And nowadays, the growth is much less. And in the recent year, in 2020, the first 250 plants were shut off. Luckily, only small plants. So the biogas production didn't go down much, or not at all, but still some old plants are shutting down now in Germany. And still we have we have a stable employment, jobs about 47,000 in the biogas sector, mostly in rural areas. And but you can see here the amount of biogas plants is going down, and that is a measurement for the politicians. Are they doing enough? Are they doing? Do we have favorable frame conditions so those plants can carry on, and can we keep the biogas production up? And that is a big challenge now, which we are facing. We have a new renewable energy law where we have to apply for tenders. This is not very good for biogas. It's very insecure. You never know whether you get the project. The politicians say biogas is too expensive. We need to lower the price. But on the other hand, they make more and more rules and regulations to make the installation more and more expensive. And if you look at the innovations we are talking today, these innovations, they do not come without a cost. And if you add such a device, whatever it is, to enhance your production, it's first a cost. And you have to see that the revenue is coming back to you. And that's a big challenge which we are facing now. And the politics said in the coalition treaty and in the new renewable energy law that we want to keep the biogas production at the same level and even increase it slightly until 2030. But they still need to deliver, because with the law which we have now at the moment, this is not enough. They think it is, but the biogas industry doesn't think it is. So the situation is like that we have a stable biogas production over the last year. The capacity increased. We do flexible production. That means we produce electricity on demand, and more biogas is going into CNG and LNG. That's a situation now in Germany and in other European countries, where also the subsidy schemes are promising lower prices, lower support. In France, for instance, they introduced last year in November a new law there, whereas until 2028 the biomethane production needs to become cheaper. So there's a lot of demand that we produce more and more gas, but less and less money is paid, and there's a big pressure on us. So I now want to go into some technologies which might be more efficient. And there's a very old technology. It's the garage type digester, which was actually developed in France. Now the Germans have taken it on about 20 years. And it's still a very good technology. It offers cheap operation because there's not many moving parts in there. That might be for some projects something to look into. and the biomass is just irrigated there in the middle, and it's all gas tight, of course. Then we have hydrolysis is also an option, and I think a colleague of mine just now will talk about it, an enhanced hydrolysis, where we have a further step. And Jean-Philippe was talking about different stomachs today. So here we have different stomachs here for the biogas project. And there we can also integrate a hydrolysis into a system to digest difficult substrates which need to be pre-treated. And there are different hydrolysis concepts, but everything needs to be controlled here very well. And also one thing of innovation is that there are new developments in intelligent control systems. We are also joined a company which is offering intelligent control systems. They do predictive predictions, and it's artificial intelligence incorporated in there. That's quite exciting that biogas is going that route. Of course, control systems also cost some money, but they also promise higher efficiency. So that's also a practical route, sort of taken from science into the practical world. And of
Ortaya
course,
koymuş olduğu yenilikleri pratiğe uygulamanın örneklerinden bir tanesi bu. Biyogaz yatırımlarının durumuna da bakacak olursak birkaç yıl önce bir
investment is going down to a certain level, but then afterwards it's not going down anymore. The specific costs are going up again later on with bigger plants. So this we have to see, but we have to know that operating costs make most of the cost of the total life costs of a biogas project, 80%. So we have the depreciation cost, the interest charge, the maintenance and repair, the maintenance and repair of the upgrading plant or the engine, the insurance and labor costs, the cost for the substrates, and the cost for land spreading. So this all has to be considered, and 80% is quite a bit. So there we can really save money. And how can we do that? Now we're going to some innovations, and during this day my colleagues are talking about more here process optimization. And one thing is we can inject hydrogen gas into the digester to produce more biogas. This is already practically done. About 30 plants are doing that already with success. It's a small box here and an injection point where hydrogen is sort of injected into the digester. And here, this is an electrolyzer. The electrolyzer is run on the electricity from the biogas plant, and then the biogas production through that is enhanced by more and more gas. So it's somehow paying off. We can also go and say, well, let's do some low tech. Let's do some low tech. And there have been some developments going on as well to save costs with a simple chute here for solids. You just push them under the liquid level. It's a rather exotic feeding system. It's used a lot in digesters in third world countries, in household digesters, just translated into big digesters without any augers, without anything. Even it doesn't need to be chopped very well. There are no moving parts. And Yeah, well, it must not be empty because otherwise you have CH4 emissions. So it's covered here, and the digester has no mixers, no heating inside. It's all done outside. There we have a phase separation, and the phase separation means that there's liquid on the bottom. So the liquid is pulled out, run through a heat exchanger, and sprayed on top with a nozzle with a modulating pump. And it covers all the surface of the digester and sort of throws the water on top. So this is a low-tech approach. The investment is relatively high, but the operating costs are relatively low. Also, one thing could be to go away from electricity production and invest into a decentralized CNG station. That's what one farmer in our area did. Instead of only producing green electricity, there is a certain readiness for this technology in vehicle to run your vehicles on bio-CNG. And there are also cars which can be bought, and those cars can then fill here at that filling station as well. And they come to the farm instead of like going to a shop or whatever, or additionally you go to the shop and then you also fill up your car. That's the idea. But also the idea is to run your tractor on bio-CNG. And how can it be done? Yes, or you also have to say, well, we also have to use renewable fuels on the farm. So bio-CNG is an option. We have here 4 gas bottles, 1, 2, 3, 4 on the other side as well, 2. So that means full day farm work autonomy. And New Holland has got some models which go on biomethane. So this is all possible already. No batteries on the farm, it's too heavy. So bio-CNG can be an option. So I'm almost finished with my presentation. I just gave you a small glimpse on what is going on in Germany. So we actually definitely need innovations to tackle end of feed-in tariff contracts. We need to do technology adaptations, combining low investment costs with high rate degradation performance. And in order to tackle the demand not to use any more energy crops, we also need to have we need to have substrates which are not so expensive, but for those we need a good solids or dry digestion in batch or continuous mode or other upgrading technologies. Thank you very much for your attention.
Okay, Mr. Köttner, thank you very much for your really interesting insights in regards to what's going on in Europe and Germany. I really find it very interesting. Let me share my screen first. If you let me do that. Then I'll try to give what's going on in Turkey. I hope you see my screen right now.
Yes, it's fine.
All right, thank you. And in addition to what's going on in Germany, I'd like to give some more information about a new market, which is Turkey. Turkey is a really new and large market for biogas sector, and the incentives, governmental incentive has started 10 years ago. But in that last 10 years, we were able to achieve the construction of only 65 biogas plants. The incentive was only 13.3 US cents per kilowatt hour as of last month. But unfortunately, starting from last month, these incentives were reduced dramatically, almost by 40%. On the average, I think it's around 7.5 US dollar cents per kilowatt hour. Yes, this is a big wound, or we are crumpled in terms of investments. It's not surprising to see a slowdown in the new investments in Turkey. But as Professor Angledaki and Jean-Philippe already underlined, I really value these extra information because we can remedy these wounds or bandage it or dress a wound with new value-added chemicals or commodities, not only biogas. So if we just trust on biogas, especially for Turkey, that's what I'm saying, because the incentives are reduced and it's not so easy to recover your money back in a reasonable time. But as already mentioned before in the morning, that we can produce many other value-added chemicals, including organomineral fertilizers, single-cell proteins, succinic acid, thermal energy, whatever you can count. So there are many other options to survive in terms of economy. So, but what I will do for Turkey, because I'm trying to lift the soul for biogas in Turkey, this is a scary scenario for Turkey because we have just started and are slowing down now because we were able to build only 65 biogas plants. Let's see the profile in Turkey, Turkey's current situation and energy profile. We use, of course, we depend on fossil-based energy sources mainly. You see natural gas and of course, we have a significant number of hydropower stations, but mainly we are depending on the fossil-based ones. If you look at the energy profile for renewables, and wind, geothermal, and the solar energy is also in the increase. So it's exponentially increasing sector. And biogas was able to join this, let me call race, but now with the reduced incentives, perhaps we will have some slowdown, as I mentioned. This is the portion of the total installed capacity in Turkey, and mainly, as you can follow from here, we're depending on the fossil-based energy sources, and the renewables has a small percentage, but this is a promising sector in Turkey. As I already mentioned before, Turkey right now has 124 units of a licensed biomass plant, and 65 of them is biogas plants. Others are the biomass burning combusting systems or landfill sites, but physically 65 of them are biogas plants, and main substrate is organic waste, including animal waste, agricultural waste, forest waste, herbal waste, urban waste, and other wastes. And in contrast to Germany or Europe, right now in Turkey, I think we're more careful about using the energy plants because of the energy and food conflicts, potential energy and food conflicts in Turkey. But we have excess amount of organic waste material that could be exploited or used for biogas production in Turkey. Maybe if we finish sometime all these organic materials, maybe we can add some energy plants.
Belki daha fazla stratejik sebeplerden biraz daha gelişme.
Contrast to Germany again, there is a big interest for the larger size of biogas plants in Turkey. And we have a fixed incentive rate. If you look at the Germany and the Europe, that incentive supports the smaller investors, farmers better. They have higher incentives. But in Turkey, we have a fixed incentive, and now it's on the average 7.5 US dollar cents per kilowatt hours. And there is no incentive for thermal energy. So, and for fertilizer side, it's up to you personally that if you're able to sell it, then you can have some additional income from that. But the main income comes from the electricity production. But as already mentioned by other speakers and by Mr. Köttner, biomethanization for biofuel production is also another promising sector. That's what I have been repeating many times in my conferences, saying the biogas investors that don't depend on the biogas for electricity production, but also add some purification stages to get some biofuel from the biogas plant, so that we can also support the Green Deal. You know, as you already know, that the Green Deal will be limiting the carbon emissions very much. So these are the numbers of the units, but as you see, mainly they are over 1 megawatt installations. And I have already mentioned that we started in 2005 with, here you can see 13.3 US dollar cents, or 133 US dollar per megawatt hours incentives, but now it drops down to, and it is now not US dollar, it's Turkish lira. That's also another problem. But on the average, it's around 7.5, which corresponds to 40% drop in supports. You can also see from this table, you see in the past, as of last month, it used to be 13.3 for biomethanization. Now it's around, on the average, 7.5. That is a big concern now for the new investors. So we will see what's going to happen for the next year. Hopefully, as already presented by the other speakers, we will draw the investors' attention towards producing other value-added chemicals and commodities like organic fertilizers and 6-unique acid and other single-cell proteins or others, hopefully. So I think this is all I'd like to share with you in regards to what's going on in Turkey, and I thank you for your attention. Okay,
Thank you very much to both of you for this nice presentation. So I think we've got one question from the audience. So the question is, as large biogas plants in Germany are shutting down, to which sector the owners are shifting to?
At the moment, there are no large biogas plants shutting down. It's only very, very small plants which have been constructed over 20 years ago. They're very, very small farmers. So it's basically an average capacity of 40 kilowatts. So it's very little. It's very small plants. So the large plants, they only will be they were only built in Germany as from 2005 to 2014, and that will still be that will still be coming, but we hope it will not be coming because politics, needs to rethink, I think, and needs to also rethink to keep what we have, because shutting down renewable energy production at this point, when we need actually more, doesn't make sense.
That's I think we all agree with that. So if the audience, if anybody has more questions, we can continue with questions from this presentation just before lunch. So now we continue with the next presentation, but just if you want to ask something else, we will do all the questions after the next presentation, just before the lunch break. So our next speaker is Carsten Smuk, Head of Research and Development at Pre Power, a recycling energy service in Germany. So the floor is yours.
Test, test. Can you hear me?
Yes, we hear you well, and we can see your presentation.
Okay. Okay, hello everyone. I'm pleased to be able to speak to you today. My name is Carsten Schmuck. I'm a bioproduct technologist, and I present today my company, the PRE, the Power Recycling Energy Service GmbH. I'm the head of the research and development department there and take care of ongoing projects. Today I have the pleasure to tell you about our patented process of combined hydrolysis and novel process configuration for enhanced biogas production. But before I start, I would like to give you a very, very short introduction to our company. We are a small, medium-sized company with 12 employees located in the north of Germany. near the Baltic Sea between Berlin and Hamburg. We have been around since 1995, so we have over 25 years of experience. We are an innovative and board-based company with many years of experience and knowledge in the biogas sector. In particular, we have specialized in the production of renewable energy and the use of biomass. We plan and project biogas plants, local heating networks, heating, cooling, and air conditioning systems, and we have a small research and development department. This is how the express hydrolyzers, the high performance fermenter, the combined hydrolyzers, and the Wavebox, and among others, were created. Today, I would like to introduce you to the combined hydrolyzers and the Wavebox. And finally, I give you a look at the project we completed in January this year. It's called the CombiMax. It is a synergy of ultrasound and cold plasma, but more on that later. Okay, let's start with our patented combined hydrolyzers. Here in this figure, you can see an example of combined hydrolyzers. This plant is located in Sarrentin, 1 hour away from our office. We have two another installed hydrolyzer systems in Potsdam and Bloomberg. Sarrentin, this plant was installed in 2016 and ran trouble-free on the part of the technology. Here at the front, at the beginning, is the feeding chamber. Maybe you can see it a little bit more in detail. This can be exceeded as needed with a wheel loader for feeding. You can see this here. After the tank is filled and the inlet sealed tight, through the co-maceration with the content of the pumping chamber, the solid substrate contained in the vessel is brought into intimate contact with the active microbial material. As great homogenization of substrate components take place under the action of the agitator. You can see the agitators here and the cutter and the pump. So the pulp material is heated to a present temperature. The pulp is now ready for the third step, the pumping. The chopper pumps removes it forcibly through a chamber separated from the tank inlet, shredding and the coarsest component in the pulp and dissolving aggregate masses, wheel pumping the substrate back to the little chamber. In the following stage, the microorganism actively promote the hydrolysis. The fermenter is now continuous feed with hydrolyzed substrate that is perfectly matched to the gas production requirements. Biological hydrolysis continues until the next feeding process begins. The gas released during filling and gas production by hydrolysis is fed into the biogas tank. So what are the essential advantages of the system? A great advantage is that the feeding and the hydrolyzers are carried out in parallel in one system. We are not limited to the specific feedstock, as the process can work with almost any inputs, maybe solid manure or poultry manure, maize, straw, grass, waste, etc. The separated hydrolyzer stage helps to increase the biogas yield, and the rule of the digester is effectively transformed into that of second fermenter stage. This makes the methanization process much faster. Existing biogas plants can be upgraded by integrating a combined electrolysis unit. Typical an upgrade makes sense when a biomass feeding plant has to be rehabilitated. Additional hydrolysis stages needed to increase the biogas yield or change of input materials to solid manure, grass, maize, or similar take place. Some of you maybe will certainly have picked up your eyes when you heard the keyword increase the biogas yield. The combined hydrolysis system can stand alone or be combined with our wavebox system. This is another research project and meanwhile established process. And this is my second topic. Yeah, the wavebox is devised for treatment of suspension of organic components in water by ultrasound. The aim is to improve the degradation of the components of the fermentation substrate down to the cell pressure in such a way that a higher energetic yield of the fermentation substrate can be achieved. The wavebox, for this purpose, we use such a high ultrasound sonotrode as shown in this picture here. The ultrasound, we work with operators in the frequency range beyond audible sound. That means from 20 kilohertz to the megahertz range. In aqueous medium, ultrasound wave cause periodical compression and expansion of the sonicated medium. A high intensity ultrasound causes the water phase breakup in the negative pressure phase, resulting in the formation of the microscopically cavitation in the liquid. These bubbles will fill with water, steam, or gas. They grow in the tanks and phase and shrink in the pressure phase until they implode. This mechanical shear force generated by ultrasound are able to break up any surface, no matter how strong they are. That's how does it works. My next speaker, Dr. Nickel, I think will certainly go on into more detail on this after the lunch break. Yeah, so what are the biological and physically effects? Why did come we up with such a system? With the wavebox, we significantly accelerate the composition of substrates in the fermenter of a biogas plant. This enabled us to make cellulose material even more biobehavioral, and at the same time reduce the amount of organic material at the end of overall process. On average, we have achieved an increase in the biogas production of 10 to 15% of our customers. Yeah, and further advantage of ultrasound treatment are the reduction of the viscosity and the physical chemical stability of the fermentation material. Yeah, is improved. In detail means this means the reduction of agitation time in the fermenter and digester store shorter pumping times, reducing wear and repair costs. Our customers include biogas plants of all sizes. At the moment, we have installed 14 boxes this were connected to our system, the Wavebox, to their biogas plant, sorry. What you can see here on this chart is a letter of confirmation from our customer. Stadtwerke Neustrelitz is one of our customers. Here we are told after 6 months of Wavebox installation, they have achieved saving in daily feedstock and an increase of the biogas production. Another example is the installed Wavebox in the summer 2018 at the biogas plant in Sandhagen. You can see the biogas plant here, and this is our Wavebox. This plant has only kettle slurry as feedstock. The average residence time in the digester is 37 days, and the room load is 1.81 kilograms organic dry matter per cubic meters a day. Originally, the plant was designed for an installed electric power of 250 kilowatts. After a short time to become clear that it was not sufficient for the quantities of the kettle slurry produced, and the plant was increased to 370 kilowatts with a more powerful CHP unit. However, this were never exhausted. The installation of the Wavebox increased the average rate from 272 kilowatts to up to 317 kilowatts. The result was an increase of 70% more electric power and with an increased rate of biogas production by 40%. And again, we have received a letter of confirmation from the plant operator, which you can see here in this figure. They confirm that electrical yield increase about 70%, and they have the rate power from 272 to 317. The dry matter and the organic dry matter of the digester decrease significantly at the same time. This in turn led a decrease in the viscosity of the digester, resulting in shorter stirring and pumping times. So I promised you at the beginning, I would like to give you a brief insight into our recently completed project. So short introduction for you. In the mid-2017, we launched a project founded by the European Regional Development Fund to test and build a combination of ultrasound and plasma in one system. The project was called the CombiMax, development of a combination device for the target physical modification of ingredients in suspension by ultrasound and cold plasma. Together with our project partner, the University of Rostock and the Leibniz Institution for Plasma Research and Technology. The aim of this project was the construction of a demonstrator which include both systems. In other words, a reaction chamber where both effects can act on one medium. It was hoped at advance for a synergetic effect of both processes, both chemically and physically. Well, what you can see on my next slide is our demonstrator. Here is the heart of the reaction chamber. On the left side and the right side are our two ultrasound sources and sonotrodes, each with a power of 1 kilowatt. here a little bit difficult to see, maybe on the next picture, yeah, is the plasma source, this one, this black one, and this is the waveguide. This is a microwave plasma and a coupler extended out of the demonstrator where the plasma discharge take place. For a better understanding, here the Hue again represented. The ultrasound source are here, and this is the coupler. And before I forget, this is the inlet and this is the outlet. I was able to film the discharge in a test solution. I hope you can see it. Later in the fermentation substrate, unfortunately, you can no longer see anything, yeah, but you can hear the sound of the plasma. It's kind of like a machine gun, yeah. Okay, it also worth to say that our project partner, the ENP Greifswald, succeed for the first time to igniting a plasma in a liquid. No one has ever succeeded doing this before. And now I have brought a test evaluation for you to look at. The sonicated medium was here the kettle slurry with a dry matter content of about 4%. This was a batch test, or another word for fixed test. The demonstrator was filled and then initially sonicated with ultrasound. After the test, the sample was taken. The demonstrator was emptied and filled with new fermentation substrated and treated with plasma. And the procedure was repeated, and in the third run, both procedure was used in combination, so the plasma and the ultrasound. The x-axis shows the sonication time, and the y-axis here shows dissolved chemical oxygen demand. This is one of many parameters that we have studied ourselves as PRE. Many of you cannot do so much with this measurement, so this value is a measure of sum of all substances presented in the fermentation medium that can be oxidized under certain conditions. Increasing this value by treating the fermentation material with ultrasound and plasma in combination leads to additional gas additional gas potential. One of our process now has now been the proof that both processes in combination, the dissolved oxygen demand goes into solution significantly faster. You can see it in this graphic, and even more in the medium. Normally we get a digestion degree of 10 to 15% with our wave box, I told you. Depending on the medium, maybe even more. Yeah. With the new design and the sonotron arrangement and the plasma source combination, we were able to achieve biomass digestion rates up to 30–34%. This was later confirmed in further batch trials and industrial related conditional trials at a biogas plant. Our project partner, the University of Rostock, has investigated other parameters in addition to the COD level, such as the viscosity, the organic dry matter content, the fiber digestion, the pH value, particle size, and many more. Unfortunately, sorry, I did not get the release for the publication from them. Yeah. However, I would like to mention one important thing. In my test on the demonstrator, I noticed a significant increase in pressure when I treated the biomass with ultrasound and plasma in combination or only with plasma. In the last phase of the project, I tried to find out what are the facts about this, and the result was I found out that a gas was being produced. To do this, I built a device, shown here, to collect the gas in such a gas bag. Also, the gas bags were sent to the University of Rostock and analyzed there. And initial results indicated that hydrogen quantities of over 5,000 ppm could be obtained by treating ultrasound and plasma in combination. Unfortunately, I could not find out the exact amount. But I'm still working on it. The reason is that the project funds has been used up and the time was over. But we are already working on a follow-up project in the future. Maybe we find this out. So in summary, I can say that we reached the research goal of further developing the reaction chamber. We have been able to advance the increase of the biogas yield and degradation rate, further the increase of the biomass fermentation efficiency, and we optimize the energy use. We are able to accomplish the destruction of hazard organic components in the wastewater, processed water, and drinking water. We improved the sedimental removal, very important for the maintenance later, and reduced sonotone and plasma wear. For further questions, please feel free to contact our company or me. And my last words is thank you for your attention.
Okay, thank you very much, Carsten, for your presentation and for speaking so well to the time scheduled. There's one question from the audience, although I think you already at some point answered it, but still, there's one question saying, what do you think about microwave? Can we use microwave instead of ultrasound? And do you have any work about this?
Okay, the microwave we use is cold. The plasma we use is cold plasma and produced in form in a microwave. The microwave plasma. This one question I see.
Yeah, I think the question.
We use the microwave and the ultrasound only in the project together in one chamber, not in the real phase or on a biogas plant or something. Not only in the project.
And I think the question refers, do you think that in the future or it could be used instead of ultrasound, the microwave?
No,
or not?
No, differently.
Okay, so is there any question from the audience for any of the speakers of the session, this first session of the workshop? The question is, we have a new question, which is the specific energy consumption for the Combi Max?
Now we find out that this is much lower than the consumption of our wave box.
Any other question from the audience? For any other of the speakers of today? Okay, so thank you very much to everyone. And if there are no further questions, then we leave it for now and we have the lunch break and come back in an hour. So see you then.
Thank you. Bye-bye.
Good afternoon, everyone. I think you can hear me.
Yes.
Okay,
Yes,
that's good. That's good. Hello. Good afternoon again to dear participants. Let me open the second session of this very valuable workshop. I would like to thank to the organizing committee, especially to Professor Azbar. I'm also very glad to moderate this second session. As I see, we have very interesting three presentations, talks. And let me introduce myself. I'm Professor Barış Çallı from Marmara University, Istanbul, Turkey. I'm a professor in Environmental Engineering Department, and I basically work my research interests are on
Ve temel olarak benim çalışma alanlarım ve uzmanlık alanım
regarding biogas technologies and resource recovery from waste materials. And let me now introduce you the first speaker. He's already on the screen. The first speaker is Dr. Klaus Nichel, and he's CEO of Ultra-Waste Water and Environmental Technologies in Karlsbad, Germany. His company is one of the international market leaders in ultrasonic biomass disintegration. Dr. Nichel acts as consultant for all methods related or regarding treatment of wastewater and biomass processes. And also he is an expert in biogas disintegration and developer of a unique high power ultrasound technology for disintegration of biomass. And the title of his presentation is Effective Use of Sonication Technology for Enhanced Biogas Production and a case study. So he will talk about the case studies. So Dr. Nichel, I want you, if possible, to finish your presentation in 30 minutes. And the last 5 minutes, we can give a chance to the participants to ask some of their questions to you. I will read the questions from the chat and I will take some notes and try to summarize. Please try to ask your questions in English. Even I can also translate them to from Turkish to English, but if you write them in any other language, I don't know them very well, so maybe my language may not be enough to translate them from German or any other languages. Please try to write them, ask them in English. So now that's the time. I have already used the 3 minutes. Sorry about it. I will give you that extra minute, Dr. Klaus. And the platform is yours, please.
Thank you very much, Professor Baris, for the kind introduction into my presentation. I will share the screen. I hope you can see it now.
That's okay, but only the full screen or the slideshow, please.
1 second. Presentation. It's okay.
Okay, that's okay. That's
okay. Yes, as you announced, the title of my presentation is Effective Use of Sonication Technology for Enhanced Biogas Production. I will give you some case studies. First, a short background. We are a spin-off company from Hamburg University of Technology, and we have done 25 years research in that field with a big research group, and we have published more than 100 articles about these topics, and so we have a fundamental know-how before we went on the market in that application. Please let me give you a small overview of my presentation. First, I want to give you a small introduction into the facts of ultrasonic disintegration of biomass. Then I will step into the topic of enhancing anaerobic biomass digestion on wastewater treatment plants. Here we speak about, I speak about sewage sludge treatment. Then I will give a focus on enhancement of anaerobic biomass digestion on farmland biogas plants. As you might know, in Germany, we have 9,000 farmland biogas plants as a strategy to produce green energy. And finally, I will give you an information about the development of a suited so-called high-power ultrasound reactor or high-power ultrasound system. Ultrasonic disintegration of biomass, what does it mean and why do we use it? And this is a general schematic here, what happens in an anaerobic degradation of any organic material. You have a fermenter, anaerobic fermenter, which uses a high retention time, very long digestion time. And you have a certain degradation of the organic material. And this depends on the nature of the organic material and also on the digestion time. Digestion time is a hydraulic retention time in the digester. And our goal is at a given retention time to increase the volatile solids degradation, which means to produce more biogas. And another goal is also to reduce the retention time to a shorter retention time so that we get conventional volatile solids degradation, conventional biogas production at a lower retention time to make the investment into a new anaerobic digester smaller. What happens if we sonicate biomass, if we disintegrate biomass? We have developed a kind of two-step model. What happens if you put first energy into the biomass suspension? The first effect is the deagglomeration of the biomass agglomerates. You have a smaller particle sizes. We determine this with particle size distribution analysis with laser technology here at our company. And so the second step, if you introduce more and more energy, if you put more and more energy into the biomass, you have a solubilization of that material. You can degrade, you can open up bacteria cells, and you can also open up, depending on energy, you can also open up some structures and release some particulate COD into soluble COD. Carsten Schmock showed you late in the previous presentation. You determine that degree of disintegration by the analysis of the soluble COD. So you transfer particulate COD into soluble COD, and this is much better biodegradable. This is a light microscopical view just to show you, to visualize you what happens if you sonicate microbiological biomass, which is sewage sludge. You see with a short sonication time, you see a complete homogenization of the biomass. And with a longer sonication time, you cannot see a big difference in the light microscopical analysis. There you need to determine the increase of soluble COD. And this determines the degree of cell disintegration. Enhancing anaerobic biomass degradation on wastewater treatment plants. We are coming from the, as I mentioned, from the Hamburg University of Technology Department of Wastewater Treatment. And we have done 25 years research. Part of our research was done in a small scale. Part of our research was done in the pilot scale equipment. And here you see our pilot scale equipment, which is consisting out of 5 anaerobic digesters in 200-liter scale in a container system which we could bring on several wastewater treatment plants to do pilot scale experiments. And this plant, in that plant, we can really set up a full mass balance. Because on the full-scale applications and the case studies, which I show you later, it's more or less not possible to show you full mass balances. A lot of things changes, but here we have at least one digester which was operated in parallel as a conventional system, and then you can directly compare the degradation of disintegrated biomass with the conventional degradation. You can set up 100% mass balances on that scale. And on that experiences, we have done also a lot of publications because they are very, very solid results came out of these experiments. And just one set of our experiments which are shown here. We have sonicated the biomass before it entered to the anaerobic digesters, and we operated at very short retention times, reduced the hydraulic retention times from 16 days to 4 days, which is very, very short. 4 days is still the risk to wash out the methadogenic bacteria, but this is still possible in a CSTR digester. And we compare this with the conventional degradation at 8 and 16 days hydraulic retention time. What happens, just to show you an example, if you have a conventional degradation with a shorter retention time, you have a lower volatile solids degradation, and with a higher digestion time and hydraulic retention time, there's an increase of volatile solids degradation depending on the organic matter which you feed. And with the sonication, with the ultrasonic pretreatment, you can improve the volatile solids degradation. Especially at the shorter retention time, there's a relatively higher improvement of the volatile solids degradation, which also means a higher improvement, relatively higher improvement of the anaerobic biogas production. And another important topic is, or item is, that we can achieve also at very short retention times volatile solids degradations in the range which we conventionally achieve at much longer retention times, which shows you that we can speed up, accelerate the process tremendously. We also did kinetic analysis. We put this all in the first order anaerobic reaction kinetics, and we saw that the K value of the anaerobic biomass degradation can be speeded up by a factor of 10. So it's a tremendous acceleration of the anaerobic degradation process. In other words, if you construct a new anaerobic digester, you can make it much shorter. You can design it to a much shorter hydraulic retention time. So case studies, full-scale experiences. I speak about long-term experiences. This is a wastewater treatment plant now in Germany. Bamberg is one of the biggest Bavarian wastewater treatment plants, and they are have a typical anaerobic digestion of the sewage sludge. 90% of the sludges in Germany are treated anaerobically because you want to use the biogas, and you have also better dewatering characteristics of the anaerobically degraded sewage sludge as compared to aerobic stabilization. The design capacity here is 220,000 population equivalents. It was an overloaded stage when we were contacted by the plant operated, and they have typically primary sludge and waste activated sludge separately thickened. And they have 3 anaerobic digesters with an 18 days digestion time. And the volatile solids degradation was quite poor due to the fact that they have a lot of waste-activated sludge. Waste-activated sludge is a part of sludge which contains the bacterial biomass, and the bacteria are not well biodegradable. And due to the fact that they have more waste-activated sludge, the volatile solids degradation in the conventional stage was quite poor. And when this plant manager came to Ultrawaves in the year 2002, there was a conventional plan to improve the volatile solids degradation, to improve the biogas production by adding a new anaerobic digester. This is a standard solution. They wanted to add a new digester, 3,000 cubic meter X-shaped sewage sludge digester. Investment costs for that building would be 1.5 million euros, and to extend the digestion time from 18 to 25 days. But then they tested ultrasound technology, and they gave up this original plan, and they finally installed Ultrawaves high power ultrasound technology. What happened in year 2002? We made a demo test. In year 2004, we installed the ultrasonic units here, 2 ultrasonic units. This is a sound insulation cover. You cannot hear the ultrasound. This is beyond the human hearing range, but you can hear the strong noise of the cavitation. This is a very powerful effect, which is solubilizing the biomass in a few seconds, and we can increase the soluble COD by factor of 10 due to this high power ultrasound treatment here. And we just sonicate a fraction of the thickened waste activated sludge before it enters to the digester. What happens? This is a full-scale observation. Of course, things are changing from year to year, but we just observed what happened on the wastewater treatment plant. And this is, yeah, over the first years, there's a evaluation of feeding of the amount of fat sludge and the volatile solids degradation, and the volatile solids degradation went up tremendously. So I would not claim now from 34 to 50%. This is an individual case, but we see a strong effect. Even at a higher load, there was a higher volatile solids degradation. The biogas production was tremendously increased, and finally, the operator gave up to construct a new anaerobic digester. They run the system now. The operating conditions on the biomass digestion is 12 days hydraulic retention time. They have a very high biogas production, and so they can save a high amount of euros, and they did not had the need to install a new anaerobic digester. On these bigger wastewater treatment plants in Germany, they make energy balances because energy is a big issue in Germany, as you might know. We want to come soon into energy self-sufficient stage here. CO2 emissions should be cut down, and so in the municipal wastewater treatment plant is the highest energy consumption place in a municipality in Germany. And the issue is to reduce energy consumption on wastewater treatment plants as much as possible. And on Bamberg wastewater treatment plant, they make energy balances, and on that plant they look at the energy production, which is a pink curve, and the energy consumption for wastewater treatment. And this is a scale here from 2004 to 2016. And important is the percentage of energy recovery by the biogas incineration from sewage sludge treatment. And the biogas, the percentage of energy production as compared to percentage of energy consumption was, before we use the ultrasound technology, it was roughly 65%. And then it increased steadily. And from the year 2010 on, they installed also new gas engines on that wastewater treatment plant, new combined heat and power plants. And this plant is operating energy self-sufficient. So in other words, they produce so much biogas by the anaerobic stabilization of the sewage sludge that they do not need to purchase any extra energy. This is energy self-sufficient, and they can use the electrical energy for the complete wastewater treatment operation there. So they are energy self-sufficient from the year 2010 on. And we made an energy balance for the ultrasound equipment itself, and the energy input to energy output, energy gain of that technology was 14 times more electrical energy by extra biogas production as compared to the energy consumption for the high power ultrasound system. So it's a very high efficient system for that wastewater treatment plant. So second topic which I touch is the enhancing of the anaerobic biomass degradation on farmland biogas plants. As I mentioned, we have a strategy here. We had installed 9,000 farmland biogas plants in Germany as it's a strategy to produce green energy. And when we started with our R&D work in the farmland biogas applications, we searched for a plant with two parallel streets of treatment so that we could introduce our high power ultrasound technology in one of the line and compare it with a conventional line. And we found a plant which is called Bordesheimer Land farmland biogas plant, and they have two parallel lines, each 540 kilowatt energy production, and each line has the same configuration: a main digester, post digester, and a storage tank. And in each line there was just one feed, no feed mix, so that we could do really a comparison of high power ultrasound treatment with a conventional degradation. Retention time 90 days per digester. So, and the biogas production was 6,150 cubic meter per day. The goal on the farmland biogas production is the farmland biogas plants is to reduce that amount of substrate, because they have to pay a lot of high price for the feed. And here they use maize. You can calculate with 35 euros per ton of maize. And here we did the comparison with an ultrasonic unit. When we started our research, we put it in one line. We recirculated from the post-digester in line number 2 a certain amount of biomass and fed it back to the main fermenter of the line number 2. And this is in the same range of the feeding load of the day. We sonicated biomass and fed it back to the main digesters. We always use, in the farmland biogas applications, we use ultrasound technology to sonicate, digestate and feed it back to the main digester, because the solids concentration is much higher as compared to sewage sludge, and the viscosity is much higher. And this is why we need to sonicate the digestate here and feed it back. The result on that plant was as follows. We compared it. We have 2 streets, and the 2 lines were operated in parallel before all the time. And then we started to with the sonication of the digestate, and we saw that we could reduce the substrate in the line number 2 by 2.2 tons of maize, and we also increased the methane concentration in the biogas. This is due to the fact if we open up protein materials, then we also create a higher methane concentration in the biogas during the degradation. And there was also an increase in the energy output, electrical energy output of the line number 2. The return of investment time, RE time, was roughly 3 years, and the operator invested the high power ultrasound technology for the 2 lines. This was the first step where we did a direct comparison of these farmland biogas biomass applications. Development of the high power ultrasound reactor. As I mentioned, we did 25 years research at the university in Hamburg. And what are the challenges of high power ultrasound? High power ultrasound is not a new topic, but there's a big challenge to apply it to high volumetric flows. And we wanted to achieve a high degree of biomass cell disintegration. And also we are speaking about biomass treatment with a variation of biomass properties, and there are also a lot of impurities into the biomass. So we did a lot of experimental work here and developed later a high power ultrasound system in a group of 25 people of doing a PhD and master theses at the university. This was our first system. We had, these are the so-called sonotodes. High power ultrasound means low frequency, high intensity ultrasound, which is extremely efficient. And then in a few seconds, when the biomass is pumped here through these cavitation zones, there's a complete solubilization. And the sonication dose could be reduced from formerly 30 to 90 to today 3 to 9 kilowatt hours per cubic meter with the same effect of cell disintegration. And for this technology, the university was awarded with the Innovation Award of the German Ministry of Research, and Ultrawaves has achieved the Innovation Award of the German industry, and the manufacturer was awarded with the Innovation Award of the federal state of Baden-Württemberg due to the fact that we had tremendously reduced the energy demand for cell disintegration. Sonotronic is our mother company now. We founded Ultrawaves 20 years ago as a spin-off, and this is the headquarter building of Sonotronic. This is a high power system, called oscillating system, and just some information. They have 350 employees. They are experts in the low frequency, high intensity ultrasound. They are the world market leader in these applications, and they have a turnover of 56 million euros during the last year. Later, we developed together with PRE, Carsten Schmock gave you already a presentation, the Wavebox system for the farmland biomass applications. This is a plug-and-play system. And you see here some development stages. You see very, very compact systems. These are the sonotode types. This is a pump. There's a PLC on the background. It's very, very compact due to the fact that ultrasound does not need build in big reactors. It's a small effect. It's in a small room. You create a high power ultrasound, the cavitation. You cannot bring it in big rooms, so they can make it very compact. And this is an installation here between post digester and main digester. So this is the typical application. You have seen from Carsten also the Wavebox model 2021. They combine it, PRA is combining this with plasma technology. And this is a model here in current model. And this is a process room in the control cabinet and the back side of the system. Some experiences about the Wavebox. Carsten gave you some case studies. I give you some other case studies. This is Farmland Biogas plant 17, 500 kilowatt electrical power production. And here they treat digestate and feed it back into the combi hydrolysis. And by this, we also hit some bacteria cells. And if we hit some bacteria cells, solubilize some bacteria cells, just a few active bacteria cells, we also increase enzymatic activity. We release proteins from intracellular compounds, release it, and then we have also a higher effect in the hydrolysis process. So it stimulates the overall process. We made under the university, we determined in that loop of sonication some activities of key enzymes like dehydrogenase activity, and we could see that it was in these digesters where we put it into this loop of digestate sonication, we could increase the dehydrogenase activity by factor 7 to 10. So there's also not just a kind of solubilization, it's also kind of stimulation, enzymatic stimulation of the anaerobic degradation process. Some info here about the Wavebox on 17. They compare two parallel Farmland Biogas plants, 17.1, 17.2. The maize silage could be reduced by 3 to 4 tons. The grass silage could be reduced by 2 tons per day. There was a much lower viscosity, much lower solids concentration in the digestate due to the higher volatile solids gradation. The methane concentration increased, and the return of investment time was quite short. And also the operator gave a reference letter that it's a very, very efficient system on that plant. Another second case study is the Wavebox on FBP Göritz, Farmland Biogas plant Göritz is a 600 kilowatt electrical power. Here they treat the second step digester material, the digestate from that second step digester, post digester, and recirculate it into the main digester. And the outcome here is also 3 to 4 tons per day reduction of the maize silage, the grass silage by 4 tons per day. And fibers were degraded. So in the final use of the digestate, there was no need of separation any longer necessary. The biogas, the methane concentration in the biogas increased. And there's also the operator gave a reference letter that the return of investment time is 2 to 3 years. So rather short, and the investment is highly efficient on that plant. Finally, also some slide from PRE. We compared in the research and development the final quality of the digestate, and we did some parallel tests on Farmland Biogas plants, and we saw that the volatile solids output in the digestate in the conventional biogas plant without any hydrolysis was 78%. So if we use a standard hydrolysis, this could be reduced to 72%. If you use the Wavebox, there's still a higher degradation than in a conventional hydrolysis process. You could reduce the volatile solids concentration, volatile solids content by 2 to 67%. And if you use the combi hydrolysis of PRE, it's in the same range, the performance. But if you combine the combi hydrolysis with the Wavebox, there's a highest degradation, and the residual volatile solids concentration of is just 60%, showing the improvement of volatile solids degradation. Just finally, some general information in the wastewater and biomass market studies. There's one important global water intelligence report. This is the biggest international report on market strategies and evaluation on the market technologies, and they had own report about pre-treatment technologies, and they call it the battle for hydrolysis. And there are some leaders and challengers shown where they determine it in thermal hydrolysis, which is very, very expensive. You cannot compare it to mechanical or ultrasonic disintegration. This is 10 or 50 times more expensive. So now, it makes no sense to apply it for any farm biogas plants. But in the field of the non-thermal technologies, the know-how and the market distribution is by Ultrawaves is shown here as the leader is in the available technologies in the world market here. We have I showed you some case studies from Germany. We have also a lot of case studies in some other countries, and our technology is sold today under several names, and this is always a technology which is here produced by Sonotronic. And we have many case studies in a lot of plants worldwide, mainly in wastewater applications. So we have installations from Brazil to Australia, so all over the world. And this shows you also that the technology is rather low in maintenance requirements. There is one wear part, and you must exchange one wear part every 2 years, but that's all. And then it works, and then it works for many years. So I come to the conclusions. Biomass treatment with ultrasound is an established technology. We have shown in several studies that it's very economic under the market conditions, at least here in Germany or in Europe. And so this has a great potential for enhancement of the biomass degradation. The energy demand and energy output, today you have more than 10 times more electrical energy output than electrical energy input into the biomass. So it's also a technology which reduces CO2 emissions. And important here in Germany, we have the German Wastewater Technical and Waste Association, DWA. I think also in Turkey, you might know it. Some consultants who are planning wastewater treatment plants, they plan it according to DWA or ATV, formerly ATV. And there's a one rules and regulations paper, biomass disintegration. And this paper, it's 100 pages, it shows the sweetest and most economic systems. And there's mentioned that Ultrawaves high power ultrasound technology is today the most economic system, most efficient system for solubilization of any biomass types. So this was my presentation. I hope I have not run out of the time. And if you have any questions, please feel free to contact me by the questions now or by email. Thank you very much.
Thank you very much, Dr. Nikhail. So you are very well on time. So it's a very fascinating presentation. Yeah, it's really very nice results of case studies, both from the wastewater treatment plants and also from the biogas plants. So now from the chat zone, I see some questions. So the first one is a little bit long. First of all, thank you for presentation. You stated that ultrasound waves facilitate biodegradation, cell breakdown. Can your technology be applied to gaseous fluids? Kayseri Sugar Factory, a member of our chamber, produces biogas from its own processed water wastes. It also produces biomethane by enriching this biogas. My question is, can the gaseous mixture be separated by using ultrasound in biogas enrichment processes? Actually, this is not directly with the application that you use, but it's about ultrasound. If you have any knowledge about it, can you try to answer?
Okay.
He's from, I cannot read the name, so it's Savaş Ertuğrul from Kayseri Sugar Factory. So please.
We have some installations of in the sugar factories and the residuals in Pakistan and in India. We have these installations where we have spent wash, for example, and this is a residual from the alcohol production from sugar factories. And there we also increase we sonicate a part of the digestate and increase the biogas production. The main goal in these countries, India and in Pakistan, is that they have zero waste policy, and they must reduce the COD concentration in the effluent of the biogas plants as much as possible. But we also have some installation. We have some case studies. I think maybe you can contact me directly, and I can send you some separate presentations about that.
Okay, I think that's better because this is a little bit different than the presentation that you made. And the question was asked from Savaş Ertuğrul, Kayseri Chamber of Industry. I think he will directly contact you with your email. So there are lots of questions. I would like to summarize. One question is there any case in Turkey? So is there any example that they use their ultrasound treatment? So let me combine it to the next question. What is the sonication process performance under wastewater treatment plants sludge according to polyelectrolyte bound, etc.? So that's a typical question by Olcay Işın. Could you briefly explain is sonication process affected to high cellulosic compound cells? So that's and again something like farm-based biogas plants. And one more question, so that will be the last one that I will get here. Thank you for your valuable presentation. Can you suggest wavebox or ultrasonication technology to enhance methane production? Yeah, that's a good question. From a biogas plant using dry fermentation, Duygu Keskin. So is there any application to high solids anaerobic digestion? Thank you. Just in 2 minutes, could you please answer the questions briefly? Okay. Otherwise, then maybe they may directly contact you via your email. Thank you.
Of course, I give my best. I give a short answer to all the questions. Unfortunately, we do not have any installation in Turkey yet, but this is an intention also of this conference here, of this esteemed conference, that of course we might want to make aware that the Turkish market is quite important, and we are in some tenders at the moment for Turkey, just for info. So so the question was, yes, some wastewater treatment plants. First, the question was of the if you have some polymer in the wastewater.
It's about the effect of polymer. So
of course, in general, I can put it into one answer. Effect of polymers. The effect of polymers. You need it for aggregation of the waste activated biomass. And it's no problem. So you can sonicate it because we want to sonicate high solids concentrations. But if it would exceed a too high viscosity, we would have a decoupling of the sonication effect. The cavitation bubbles and the medium, there will be a cushion of cavitation bubbles which will not enter into the medium. So this is the reason, if we have a very high viscosity of the biomass, that we do not sonicate the feed biomass, that we sonicate the biomass from the digester, because the viscosity is much lower, and then we have stronger cavitation effects, and more energy is used for disruption of the bacteria cells or the cellulosic materials. And so you must consider the viscosity. There's a viscosity threshold, and we determine this also. Normally we get a sample and we do some tests in the lab, and we measure viscosity. And so if there's a too high viscosity, you must treat digestate, because this is already degraded. There was a question, cellulosic structures. Of course, we have an impact on cellulose. We have also a separate presentation of cellulose. Of course, we can start to decompose that. So the structures are open up with ultrasound. If we introduce a lot of energy, we can also cut down the cellulosic molecules into sugar molecules, but this consumes too much energy. But this is the goal of our treatment. If we talk about farmland biomass applications or sewage sludge applications, we have in all cases, we have inert substances, which is on the one hand the cellulose, or it's the murine cell wall on the bacteria cell. And if we put energy into these materials, we can cut them down, and this starts to become biodegradable. But we do not put so much energy that we make it 100% soluble, because then the energy input would be too high. There's always a comparison how much I could do a lot with the ultrasound technology, but the energy consumption would be too high in that case. And we always consider that we get the maximum output of energy to the input. And this is sometimes that we reduce the energy input to a certain limit. This is sometimes the reason.
Okay, a summary, I think. Okay, so Okay, so that's I will remind it to you later. So that's, I think in summary you need something fluid. So it's as the solid content increase, the efficiency of so the ultrasonication decreases. So it's vice versa. You need to decide energy or the costs, or it's or the biogas efficiency, something like that.
From the
Okay.
scientific point
Yeah,
of view, we can do a lot, but we are now in the practical application. You can do so much things with ultrasound, but we must find the most economic solution. And if we consider this, we have today roughly 3 years return of investment time. And this is why we limit the energy input to a certain limit. And then we have the maximum energy output of the process. But this is the economic game, so to say.
Okay, thank you very much. And one last note from Professor Nuri Asbar. I think you have a connection with him. So it's he wanted me to remind it to you. Maybe you are in the same team from Ege University. So it's maybe in any of the projects. So that's just a reminder to you. Okay, okay, maybe the representative in Turkey, maybe that not directly you, but the representative in Turkey has some connections with Professor Asbar.
Yeah.
Thank you so much for this nice presentation. It's really, really very well. And then now I will pass to the second speaker. Thank you very much. I think they will do the organization now. Let me introduce the second speaker. The second speaker is Dr. Hans Ösner. Is Dr. Hans Ösner here?
Yes.
Ah, okay. Let me introduce him to you. He's an agricultural engineer, and since 1990, he is working in a State Institute of Agricultural Engineering and Bioenergy in University of Hohenheim in Stuttgart, Germany. And since 2004, he is the director of this institute. In the same time, he is adjunct professor of the China Agricultural University in Beijing, and National Center for International Research of Bioenergy Science and Technology. And he is mainly working on research in the field of biogas production and questions of bioeconomy. And the title of his presentation is Boosting Biogas Production via Trace Element Supplementation. As I told you to Dr. Klaus, Dr. Hans, so I really would like you to finish your presentation in 30 minutes, because I really very appreciated to get some questions from the audiences, because the interaction is better than just a monologue. So I think that will be better. So if you finish your presentation in 30 minutes, we will have 5 minutes for questions. The platform is yours. Thank you very much.
Okay, Professor Kali, thank you very much for introduction. Also, thank you very much to Nuri Asbar, who has invited me to participate in your interesting congress today. I will report about trace elements in the biogas process and look for deficiencies and for increasing of the gas yield. Yes, I want to show you first where I'm coming from. Southwest of Germany, Stuttgart, and the University of Hohenheim. I think a few of you will know it. Some pictures of the nice landscape in the surrounding, but also a very active industrial zone in the surrounding of Stuttgart. I have shown some brands. You will know them: Daimler, Bosch, Boss, Würth is also very well known, and so on. So we are well placed. And the University of Hohenheim is a fundamental and agricultural university. It's founded more than 200 years ago. At the moment, we have 3 faculties: agricultural science, natural science, and business and economics, with around 10,000 students. This is only one small university which is placed in Stuttgart. Yes, let's come to the biogas process. I don't want to boring you with this scheme. You have seen it a few times today already, but it's important to look for the requirements which are necessary to have an optimal process. We can see we are running in few steps to get at least the methane and the biogas. And it's very important to have a working hand in hand of microbes and of enzymes who are involved in this process. And therefore, it's sometimes necessary to support these steps and to help them to get a better transformation of organic to methane. Let's begin with an example from a batch test where we can see different substrates and we can observe the kinetics of transforming of organic to methane during, here in this case, 35 days. At the bottom, we can see straw in different crushing lengths. And we can see, for example, as example, concentrated feedstuff for animals with different feeding level. Let's have a look to this 600 milligram input in this syringe we have observed here. And we can see that the methanation is going relatively quick with this concentrated feed. After 5 to 10 days, all methane which can be produced is already transformed. What is happening inside of this liquid? I have drawn here the total volatile fatty acid content over these 35 days. And we can see the increasing and falling down again after 10 days when the whole process is done. On the other side, pH level is also important. If we have a very stable condition in our batch test, like it's in the regulations, we can have a relatively constant and stable pH level. It's going down a little bit only for 2 or 3 units, but then it's going back again into a constant level. Let's have a look to this stability or to this buffer capacity of in form of volatile fatty acid to total anorganic carbonate ratio, which shows us at the same batch this VFA TAC ratio. Let's have a look to these dark red lines and broken lines. We see again the acid concentration, which is increasing and going down again after 10 days. And we can see this buffer capacity, which is also increasing and going back to a very stable level of 0.2, which is in a very good situation. We shouldn't increase, or we shouldn't have a higher VFA TAC ratio than 0.3 in normal biogas processes. But here we have a batch system, and there we can agree a short increasing. Let's switch to practical biogas plants, to full-scale biogas plants, which have been observed over a longer time. We took samples and analyzed the acid concentrations and the composition of the different volatile fatty acids. You can see here the acid content in milligram per liter of the substrate of acidic, propionic, and longer chained acids. And we have here a very stable process without risks. This system is running at a low loading rate of lower than 2 kilogram organic volatile solid per cubic meter digester volume and day. So very low content of acids. Let's have a look to another example, another farm. We have a little higher acid concentration, up to 3,000 milligram per liter for acetic acid, but in total also an acceptable level without problems. And you can see it is an observation over a period of 1 year in this system. Let's come to another biogas farm. We can also find such values if the process is not running optimal. We can see acetic acid concentration up to 2,000, 3,000 milligram per liter, but a problem is that propionic acid is increasing up to 6,000 milligram per liter in a normal biogas digester. And also butyric acid and valeric acid are also detectable, which are normally not detectable in biogas processes. This is a very unstable and also inefficient process there. We can avoid that we don't get a good transformation of the organic material to methane, and will get also losses of methane yield at such conditions. Here some aspects and recommendations about the acid concentration which is acceptable in full-scale biogas processes. We can accept acetic acid, propionic acid, and butyric acid. You can see the shares or the concentrations, and the total volatile fatty acid concentration should be less than 4,000 milligram per liter at least. Yes, what can happen if we have an increasing the metabolites which are produced can inhibit our process and bring us problems. We don't be able to win the whole potential, the methane potential of the digest state, like we have heard also from Mr. Nickel before. Then we have an uneconomic system, and we need more substrate to get the same methane yield at the end. But what is the reason? We don't want to fight for symptoms. We want to find the problem and want to find solutions. Let's come to some aspects about this solution. What can we do if we have this increased organic acid concentration? What happens if the biological process disturbance is found? In the last 15 years, we had a lot of examinations in this field, and it was shown that, for example, trace elements are very important to optimize this biogas process. Methanorganic microorganisms have a special need for micronutrients, especially nickel, cobalt, molybdenum, selene, and also iron is very important. If there is a lack of trace elements, we can get an unstable process with risks, and it can go to the end of the biogas production and stop biogas production. The methane content normally is also decreasing in such unstable conditions. Yes, what is the reason, or what is the function of trace elements in the biogas process? They are important parts of coenzymes and cofactors. At the bottom, we can see one example of these cofactors, which has a central atom of nickel. You can compare it with hemoglobin, which has in our blood, which has iron as central atom, and here we have this nickel included. And if nickel is not available, these cofactors cannot be produced, and the process will be stopped, and maybe we have then an increasing of acid concentration, for example, propionic acid. Until now, about 7 coenzymes are known, and they are playing a very important role in the biogas process. Yes, oxidized forms are sometimes used as electron acceptors. For example, if we look to ferric oxide or manganese oxide, they are
Manganez oksit gibi mesela.
electron acceptors, and these metals are also reducing.
Bu metaller aynı zamanda sülfür toksitesini de azaltıyor.
Or with sulfur. Very important is there also iron.
Aynı zamanda demir de çok büyük önem taşıyor burada.
Fixing of sulfide in this process.
Bu süreçte sülfürü kontrol altına alarak etki gösteriyor demir.
Trace elements are stimulating
Bu eser elementlerin metanorganik büyümeyi canlandırdığını biliyoruz. Burada size yani sunumun devamında sizlere 2 tane
First examinations where we had running in a continuous digester system, laboratory digesters with a volume of 17 liters, and 15 digesters had been used for it. We run this system at a mesophilic temperature, an organic loading rate not too high of 2.5 kilogram volatile solid per cubic meter a day, and we had a hydraulic retention time of 40 days. These had been horizontal digesters, which had been fed 1 time manually a day. What had been the variance in these systems? We looked for maize silage and water. We didn't want to use manure because as we made these examinations, there had been some farmers who didn't want to use manure as byproduct, especially farmers or biogas systems which had no farm in their surroundings, so they couldn't use manure. Therefore, we made examinations with maize silage alone with water. We used some variants and added, for example, buffering additives. We added sawdust to get structure in the substrate, and we used also at 3 other variants additionally trace elements. Let's look to the results of these examinations. It was running over 120 days. And we can see here the pH value during this examination. We see 2 groups. One group, this one which is running without trace element addition, there we could see very quickly after 20 days already that the pH level was decreasing very strongly. We can accept up to 6.8 pH level to have a stable process. If it's going down more, then the process is inhibited and is not running very well. What is happening with the concentrations of acid? We can see here 2 points. The acid concentration was increasing even after 20 days. We had up to 5 grams per liter acid. And after 40 days, we had more than 10 grams per liter acid concentration. This had an influence on the gas yield and also on the methane content inside of our gas. The methane content decreases lower than 45% of methane. Let's have a look to the alternative variants which had been supported with trace elements. And we can see that the pH level was constant in an acceptable level, higher than 7. And if we look to the acid concentration, it's also acceptable, very low at the same loading rate at 50 to 300 milligram per liter. The methane yield was absolutely normal, comparable to the batch yield test at 335 liter per kilogram organic dry matter. And the productivity was also much higher at 1.6 liter per liter digester volume compared to 0.6 of the alternative example. What have we done later? We have looked for batch tests and taken unstable fermenter substrates from practical farms which had problems with low methane yield and with low methane content in their gas. And therefore, we made in our laboratory system these tests with different combinations of trace element addition. And we could see a very clear effect of different addition of trace elements on the methane yield. You can see this batch test is running over 65 days, so relatively long. And we could see clear effects. Some positive influence was increasing the methane yield for 25%. And we had even in this beginning or in this first third of the process, a very clear influence in the kinetics. And this is also shown if we look to the whole curves. Out of this analysis, very systematically, and a lot of them, we had the chance to find the optimal standards of trace element concentrations. And we defined it depending on the organic loading rate, which is running in these monodigester systems. In a minimum loading rate of around up to 2 kilogram volatile solid per cubic meter and day, we need around 3 milligram per kilogram dry matter nickel. If we increase the loading rate, the process needs much higher nickel content, for example. And this we did for at least 16 different elements to analyze which additives are necessary. And this is very important and used in all practical biogas plants which are not using manure as substrate. So let's have a look to our main substrates which are used in Germany. We have seen before from Michael Köttner that 50% of the substrates which are put into the digesters at the moment are energy crops. And let's have a look to some energy crops and the content of different trace elements. Very important is maize silage, and especially nickel, molybdenum, tungsten, selenium, and cobalt are in a very, very low level and not enough for this biogas process. The red line shows us this minimum content which we have found in this examination. So we are much under this need. And if we look to sugar beet puree, we had better conditions. There we have very good availability of these trace elements. Grass silage is also better than maize, and also this cereal plants silage has at least a low trace element supplement. Let's have a look to manures. Often manures are used as byproduct in biogas processes. In the last years, the use of animal manures increased again, but 10, 15 years ago, there was a strategy to reduce the manure input into the digesters. But if you look to the trace element content, again, these 5 important trace elements in our manures: swine manure, cow manure, solid cow manure, and dried chicken manure, we can see clearly that the trace element content is higher than the minimum we have detected during our examinations. And this is one reason why the combination of manure and energy crops are often a very stable system, and monodigestion of such energy crops brings very quickly problems in the practical farms. I want to show you another examination, which is also important. If we look to using of waste, for example, special production waste of coffee production, here we can see coffee pulp and coffee husk. Yes, it's not produced in Germany, but it's, for example, in a lot of countries worldwide. This material we analyzed here was coming from Ethiopia, and the examination was done by a PhD, Mr. Chela, coming from Ethiopia. And here we can see again the recommendation of the concentration of the different trace elements. And this broken line shows us the recommended concentration. And if we look to, for example, iron, zinc, nickel, molybdenum, this concentration is not high enough for an optimal biogas process. Let's come to the examination. It was done in the same digesters like we have seen before, also this horizontal system. And we changed during this run the organic loading rate from around 2.5 up to a loading rate of 6 kilogram volatile solid per cubic meter digester volume and day. Let's have a look to the alkalinity ratio in our process. I have to explain you before our variants. We have used husk and pulp. Pulp brings a higher methane yield. It's easier to digest than husk. The pulp is coming from a wet processing system. And we have compared always husk and pulp without and with trace elements. We can see this example at pulp, where we have a strongly increasing of alkalinity during the process after we have got an organic loading rate of around 5. This shows that the process got unstable. And if we compare it with the variants, which are done with trace elements, the green line at the bottom, there we had a very stable process without problems. If we look to the methane productivity, cubic meter methane per cubic meter digester volume and day, we can also see the same effect, and we can detect that the methane productivity was decreasing after this organic loading rate of 5 kilogram. In the case of husk, which is not producing so much gas, which is slowly transformed, there we had also detected a difference between the variants with and without trace elements, but it was running in an acceptable range, just acceptable at this moment.
Dr. Eisner, could you please come to the conclusions, the last 2 minutes, please?
Yes, okay. Thank you very much. I will conclude. For each component, it is necessary to analyze it and to look for the minimum and maximum content. It's not possible to take too much of trace elements because they are often also heavy metals. And if we have deficit of detected trace elements, then the microbiological metabolism will be inhibited and will not run optimal. The degradation rate decreases heavily, as we have seen, and the primary fermentation is often not limited. So it means our acids are produced, the hydrolysis is running very well, but the methane organic process is not running. And so our methane output will be lower. The buffer capacity will be overloaded in these cases, and this brings a lot of problems. For the process fermentation without manure addition, a high loading and high loading rates, there is necessary to have a control of the process to be sure that our volatile fatty acid content is not increasing. And if it's increasing compared and also with the Voss-Tack ratio, we have to react and to have look for the problems, often trace element deficit is a problem. And so we have to add such trace elements to get a good result. The use of trace elements can affect important advantages on process and substrate efficiency, especially if you have monosubstrates. This can be energy crops and also byproducts of different processing systems. Let's come to the next. I want to explain some aspects of our equipment. We are working in laboratory and also in practical scale. Here you can see our full-scale biogas plant with a electric power of now 350 kilowatt. It's equipped with 2 digester lines, so we can compare very well examinations which are coming from laboratory and upgraded in full-scale systems. A picture of our staff of this State Institute in Hohenheim. And I don't want to forget to remember you to this International Congress in September we are doing together with IBBK. And then thank you very much for your attention.
Dr. Reusner, I really would like to thank you. You are very well on time, so it's a little bit 2 minutes, but that's nothing. So we also started a little bit late. So a very nice presentation. Some years ago, we also studied on a very similar subject on monodigestion of chicken manure and found that some of the trace elements are very important, like selenium, tungsten, and cobalt. So they're really very well. Otherwise, if you don't add them, you cannot reach to normal methane concentrations. Thank you very much. So there is one question. If any more, I'm also waiting you from the chat zone. Okay, the question comes from Recep Dereli. So it's Dr. Dereli asks, what do you think about zero-valent iron addition? Do you have any experience with zero-valent iron?
Not especially zero-valent iron, but iron addition is always a big advantage because it is a very good element to fix sulfur, and also hydrogen sulfide is inhibiting the process often if you have a lot of protein in your input substrate. For example, if you're using kitchen waste or if you're using special byproducts.
Okay, thank you. There is no other question, but I have one. Okay, one question. My former PhD student from Kabul Polytechnic University from Afghanistan, and he asks Rahim Molay, what is your opinion if you separate VFAs during the fermentation process? In this case, will you not use trace element and you will reduce the cost? So just an opinion. So because if you don't trace elements, VFAs accumulate. And also in the previous presentations, there is a carbonate or carboxylate platform in Europe. So one of the hottest topics. What do you think about it?
I think the question is asking if it's possible to win volatile fatty acids, for example, as a base for platform chemicals. I don't think this is a good idea to do it out of the biogas process. You can do it out of hydrolysis, or you can do it in the silaging process. We did also examinations in the last years to increase the concentration of lactic acid, for example, for polylactic acid production, and to extract this lactic acid. This is possible, but I didn't think it's a good idea to take it out from the biogas process, because these acid concentrations during the full biogas process also inform us about unstable processes and inhibitions from one digestion step to the next. So I would separate it. I would put it into a hydrolysis process, yes, or in the silaging process in front of the biogas plant. This is also possible. And there are a lot of possibilities to combine, for example, this biogas process with other production units. We are also making examinations at the moment, for example, for producing of fibers out of the raw product, extracting of fibers, and the pulp, which is liquid and rich of easy digestible nutrients, is put into a high value digester, not in a CSDR digester, but in a fixed bed digester, for example, to get a very effective methane production. And the fibers can be used for different possibilities, for example, for paper production. There is a full-scale system running in the south of Germany with such a preparation of paper out of these fibers from, for example, special plants, which are not so good for digestion because they have a lot of fiber, but they are environmental friendly. So you can get a combination.
Okay, so it's high lignin content biomass, you say, maybe for other purposes. Okay, so now the second speak is over, and now according to the program, we have a coffee break. So now I think we have 10 minutes for the coffee break. So it's a self-coffee break. So it's you can go and take some tea or coffee, and we will be back here again, and it's so in 10 minutes. Okay, though maybe you are from other countries, though I don't want to say the time now. It changed from country to country. So we will be here in 10 minutes. Okay, thank you very much. Okay, dear participants, welcome back. I hope you had some coffee or tea. Anyway, now we are in the third speaker of this session. Actually, this is a German session. All the three speakers are from Germany. Now, let me introduce you the last speaker, the third speaker. Third speaker is Roland Reiter. He's a mechanical engineer. Is he here? Roland, you are here, I guess. Okay, you are ready? Okay. His key qualifications are wastewater and biogas, marketing and project management. Since 2005, he is the managing director of the company AEW Energy in Regensburg, Germany. Yeah, sorry for my presentation. So it's Regensburg, I think it's correct. So he has managed several projects for biogas plant with different electrical power capacities, accepting different feedstocks such as manure, food waste, glycerin, sewage sludge, slaughterhouse waste, and energy crops. And now he will present his talk, which is titled Digestion of Chicken Manure as Monosubstrate and Other Case Studies. Are you ready, Dr. Reiter? The platform is yours. Any problem with the connection?
I'm trying to calling to Mr. Reiter.
Okay, okay. Anyway, so now if you have any question to Mr. Reiter, please ask your questions again through the chat. And also, yeah, we can have some extra questions. Actually, the presentations.
Can you hear me now?
Okay, so that's
great.
We can hear you. We cannot see, but we can hear you.
Great. Just a moment. I want to share my screen.
Okay, let's test it first. So it's if he see it, no problem. But if he cannot, so.
Can you see my screen?
No. Okay, so something is now. Okay, okay. Only I want you to make the slideshow, please.
Does it work?
But we see your notes on the right-hand side. So only the slide is. There might be any other option. So only that's also okay. But it's we see also the next slide. So not only the active slide, but also okay, that one. I think maybe there has to be another alternative or any choice over there just to see the one slide.
Mr.
Okay.
If you are using the two screen, please cancel one of them and try to make the presentation mode again.
One of the participants says that if you re-enter again, close the file and open it again, he says it may work. So that's just a comment. I don't know why it looks like this. Actually, it's not a big problem. It's not a big problem. So we can see the slide a little bit smaller. So, but we can see now you are using two screens. It's something like that. We see both of your screens. So it's first, now I see the first slide and also the second slide.
I see, but unfortunately, I would like to make that smaller.
Anyway, okay. So now it's better not to lose time. So I have already introduced you. So yeah. The title of your presentation is actually the title that is to me is
okay.
Digestion of chicken manure as monosubstrate and other case studies. But if you would like to change the title, the platform is yours. And okay. I want you to finish your presentation in 30 minutes. Thank you very much, please.
Actually, we want to present our process for feedstock with a very high nitrogen concentration, like chicken manure or poultry manure. And I've seen a lot of poultry for egg production or meat production in Turkey, like in Afyon or in Şempiliç, and there is a big production of eggs and chicken meat, and as well you have a lot of chicken dung, and this is a great source for energy. So on the one hand, we can make energy and like electricity and heat, as well as fertilizer. But nevertheless, I will follow this structure. AV, we are two owners and two of this biogas company. We are in the business since 1996, and we founded this company 2005, so it's 16 years ago. We deliver technical equipment, engineering, and feasibility studies. What we don't do is basically earthworks, concrete works, buildings, and steel works. We deliver the equipment. So if we make biogas plants abroad, like in Turkey, the client makes not only earthworks and concrete works, he also does the cabling and the piping, and we deliver only the equipment which is not available in Turkey, like gas holder, gas treatment separators, and armatures. Regarding our experience, as I mentioned, we are in this business since 1996. We made more than 120 biogas plants in more than 15 countries. And the range is from 20 kilowatt to 3,000 kilowatt designed, as well as from 20 to 1,400 kilowatt biogas plants built. Our references are not only in Europe, also in the USA and in East Asia. So why is there a need for a new system for chicken manure? The studies show that the production for meat, especially for poultry meat, is rising. So on the other hand, the production of chicken manure rises as well. And the gas yield of chicken dung is very high. So with the conventional methods, it is like easy to digest chicken manure or to treat it in an AD plant. For example, with dilution, if you use a lot of water to dilute it and to lower the nitrogen concentration, it is easy, but the point is, the disadvantage is if you need a lot of water, the water can be costly, and on the other hand, you have a lot of digestate, and you have to get rid of it, and you need very big digesters and storage tanks for the digestate. The anastrip process is a high-tech solution, and it costs a lot of money. And if you need some spare parts, or you have to do maintenance, or you need to repair things, most of the time you need specialists from the supplier, which rises the costs as well. And the third possibility is to treat the digestate with ultrafiltration and reverse osmosis. We've seen many of those plants, but in the last 20 years, but it seems that the dry matter content in the digestate is just too high. It works in wastewater treatment plants for communities, but not with the digestate from biogas plants. So what is the difference? On the one hand, we want to lower the poison nitrogen, the ammonia in the digester. So we need we need less dilution. Not all the nitrogen is toxic. It's only the NH3, the ammonia. And actually, there are two possibilities to lower it. One is actually not possible in it's possible in laboratories, but not in real life. So we chose to lower the temperature in the digesters. We all know that the colder it is in the digester, the slower are the bacteria. So we have bigger tanks, but on the other hand, less digestate. What we can see here is how fast the bacteria work and how fast the biogas production works with different temperatures. And at the end, the gas yield is the same. So on the one hand, the point is we need bigger digesters. So we have retention times like 60 days in the digesters. And on the other hand, we have less digestate. We have a higher dry matter concentration in the digester. We need less heat because of two reasons. Reason number one is we have the lower temperature. We do not have 38 or 40 degrees centigrade. We have like 25 to 30 degrees centigrade. And we need less heat energy because there is low dilution and we have just a little additional water for dilution, which has to be heated. Our aim is a simple process, a cycle process, a removal of foreign bodies. Like we all know that the chicken manure from layers for egg production, they contain a lot of limestone because it's in the fodder for those chickens. They have to make all those egg shells, and therefore they get a lot of limestone sand in their fodder, but they don't use all the limestone in the fodder, so there's still much sand in the chicken dung, which sediments in the digester. So after 1 year, the digester would be half filled with sand, and you cannot operate it anymore. I've seen terrible pictures with digesters full with sand, which had to be emptied and cleaned. It's actually not a nice work. So we have to remove the sand in the digester, which we do with screw conveyors, and which we need to remove the solid part, the dry matter in the digestate. And one big goal is that we have no liquid digestate at the end of the process. Because in many countries, the liquid digestate is a perfect fertilizer. But the point is, in many countries, the people are not used to liquid digestate. They only want solid digestate because there is a market for this. They can sell it as compost. So we have to avoid the liquid digestate. And the last point is we need to remove a part of the nitrogen, so we can use the digestate as for dilution of the chicken dung. So about our we make this research in combination with the DBFZ. It's called the Deutsche Biomasseforschungszentrum. It's a federal institute which has a lot of laboratories, analysis, people who work in the labs and make research and analysis with us. And we do this research for 6 years now, 6 to 7 years. First of all, we had to check the influence of the digesting temperature on chicken dung, which we see here. We had 2 different samples and digested them with 3 different samples. We digested with 3 different temperatures, and we see the rising of the organic acids in the laboratory digesters, and we see the 39 degrees digester struggles the shortest time after like 18 days. They reach the peak of the free organic acids, and after this the bacteria get to the regular mode, and after 4 years, after 40 days, they reach the normal level of free organic acids of 40 milligram per gram or 40 gram per liter. The bacteria which are in the sample of 30 degrees centigrade, they need a little longer, so for them it takes about 30 days. And the one which get only 25 degrees centigrade, they needed 40 days in order to get used to it. But from then on, we
Alışıyorlar.
see
Ardından da
acid is eaten, and the gas production was just the same. As I mentioned, the dung in the chicken manure from layers contains a lot of limestone sand. So we equip our digesters with these screw conveyors. Our digesters are also equipped with submersible agitators, which keep the content of the digesters flowing, and the sand which sediments on the bottom of the digester begins to move and falls into these pockets. And from there, the screw conveyors take them out. So we have no more problem with the sedimentation of the chicken dung sand. In order to use the digestate for dilution, we have to remove all the dry matter, which means if we need it for dilution, the digestate must have a very, very low dry matter content. So we reached between 2 and 3% dry matter after a two-stage separation, first with a screw separator, which removed most of the dry matter. And afterwards, we added flocculants, like polymers that make flocks out of small solid parts. So we removed it with this centrifuge or decanter. And this way, here we see it much better. This way is, we see the two stages. First of all, sorry, we also used as for the second stage this belt filter press, which has a big advantage, which is it's not sensible against the rest of the limestone sand. Nevertheless, there is still some sand after the separation, and if we use if we put it in centrifuges or decanters, it can cause big damages, because the sand can grind these steel screws in the decanters, which does not help, which does not happen in the belt filter presses. This shows a trial with flotation of the digestate. First, we added these polymers, these flocculants, but the reactions of our digestate were very different. Like here you see it, some of the flocculants make a little foam, some of them make much foam, and some of them make so much foam that the whole experiment was destroyed, and we had to do it again. So nevertheless, the question is how to remove the nitrogen out of the digestate. So we got the digestate, we separated the dry matter. Now we have a dry matter concentration in the digestate between 2 and 3%. But
Mr. Reiter, you have 5 to 6 minutes left, so I would like.
Oh, really? Okay, thank you. So we chose a very simple system, which is used all over the world in wastewater treatment plants for municipalities. The point that I make it a little bit faster, if you allow. So we have, this is a scheme of a biogas plant with digestate treatment. Here you can see all together what I just mentioned. So we have here like a buffer tank. We can add slurry if we want. Here is the chicken dung feeding station into the digester. Then we get the digestate, the effluent of the digester out here into this separation. So we get, on the one hand, we get this solid digestate, and on the other hand, the liquid one with about 2-3% dry matter we get in this tank. This tank is a ring-in-ring tank, which means it's one big tank with 2 other tanks inside. We put it first in the permeate tank, which is a buffer tank, then we put it in here. which is the nitrification, denitrification tank with aeration. So first we do aeration, and then we do just agitating without aeration. In this way, the nitrogen gets out as molecule nitrogen. As we all know, our air we breathe consists of 78% nitrogen, and so this nitrogen does not influence the quality of our groundwater or drinking water. And this way is a very simple one, a very simple technology you have in your country anyway. Every country has this technology. You can make it on your own. And the effluent, you can use this blue effluent, you can use for dilution. So you can dilute the solid chicken dung. This is how it would look like in practice. There is the storage for the chicken dung, digester, post digester. From the post digester, it gets to the separation unit. From the separation unit to the permeate tank, which is this ring. Then it gets into the nitrification, denitrification tank with aeration. And at last it gets into the clarifier tank where the sludge separates from the clean water. You can see it very good here. The settlement tank is in the middle. The sludge settles down, and the clear water, or the cleaner water, is at the top and gets removed from there. The advantages of our system, it is just in short words, it is cheaper, it is simpler, and it works. That's the point. And so it's a stable biochemical process. The same energy yield, the same biogas yield, like with higher temperature. We do have this sediment discharge from the digester. We do not have to dilute that much, so we save digester volume, and we can combine it with a warmer hydrolysis. Lower energy demand, lower personnel and operating costs, sustainable worldwide proven method, and last but not least, only solid digest state. So nothing now bother with a liquid digest state you don't know what to do with. The input material was like this. For the chicken tanks, developments, references. This, for example, is the biogas plant we built 3 years ago in Kahramanmaraş. And what we see here is 2 digesters, post digesters, the co-generators, and the substrate was organic municipal solid waste. So the client gets municipal solid waste, separates the organic and the inorganic fraction, and the organic part gets into our biogas plant. That's a biogas plant for sewage sludge from wastewater treatment plants. So if you have a wastewater treatment plant in your community, you can take the sewage sludge and make electricity with it. This plant in Hungary, near Budapest, has an electrical power of 1.4 megawatt. So that's it. Thank you very much for your attention. And I hope you have many questions.
Okay, thank you very much, Mr. Reiter. So it's a very nice presentation. So it's you are really on time. It's we still have some time for questions. I am checking the chat, but it's I don't see any question, but it's I'm still waiting. So let me ask you one question. Do you have any example of such a system or approach in Germany?
We implemented right now at 3 different existing biogas plants in Germany.
Together with you remove the nitrogen with nitrification and denitrification from digest state.
This we still make in the laboratory. What we implement in Germany, we remove corn silage as a substrate with chicken dung, and subsequently we lower the temperature in the digester as required.
Okay, because of ammonia inhibition to
right.
Okay.
Right.
Okay. Any questions? So I check. What about the other nutrients? Do you have, or in your case, I didn't see any resource recovery, because with nitrification, denitrification, we remove the nitrogen and convert it to the nitrogen gas. So we release it to the atmosphere. What about the valuable plant nutrients like phosphate, potassium, and etc.? So it's they will be in the effluent, right? So there may be an also accumulation if you recycle it again and again to the digester.
Right. The phosphate is always connected to solids. So most of the phosphate is in the solid part of the digestate, which I just get to this picture back. So
yes,
most of
them in the solid, but it's about 30 to 40% probably is in the liquids.
Right, which doesn't bother.
Okay, no inhibitory, but in the discharge, do you have any problem related to the discharge of the treated digestate, the liquid fraction? Because in nitrification, denitrification, phosphate stays in the system, so it's
right. We haven't recognized any problems yet. We also use this biophosphor removal, which means we keep the bacteria part of the time in anaerobic phase, so they take more phosphor than they actually need, and afterwards they are in the solid part of the digestate, and we haven't recognized any inhibition by potassium as well.
Okay. There is one question. This is not directly related with your presentation, but if you would like to answer, I would like to ask you. So it's Yasin Sarsh. So he asks, do you have any experiences related with the effective microorganisms used in biogas plants, or to enhance the biogas production, effective microorganisms, some spatial microorganisms? Do you have any experience, or is it something out of your expertise?
I'm sorry, but I didn't understand the questions.
Effective microorganisms, some spatial microorganisms. Do you have any experiences with the use of effective microorganisms, adding some extra spatial microorganisms to biogas fermenters from outside? Do you have any experiences?
No, we figured out that actually the microorganisms are in the sludge anyway, and there is this antagonism, and the group of microorganisms which are adapted best for like this temperature and this feedstock, they get a higher population, and there is a special methanogen archaea which are able to handle higher nitrogen concentration, and you can see them in the microscope with ultraviolet light because they are luminetic. They get a higher concentration. This is a higher concentration of nitrogen is an advantage for them, and so they are like ammonia feel.
Okay,
and
so it's spatial and natural selection in the digester. So the stronger ones adapt to high ammonia concentration. What about the treatment part? So the treatment of the digestate, the liquid fraction of digestate.
Yeah.
Do you suggest to use Anamox process? So it's instead of nitrification, denitrification. Do you say? Would you like to say something about the Anamox process?
Yeah. We are very optimistic regarding the Anamox process. We make in the lab, we make regular nitrification, denitrification. The Anamox process, the big advantage is that it needs less aeration and for sure it needs no additional carbon source for the denitrification. So actually, as soon as we checked the nitrification, denitrification, we will start with the Anamox.
Okay, thank you for your presentation and also the answers to the questions, Mr. Reiter. Thank you very much. And now, the dear participants, this is the end of the second session. Now I would like to close and give the mic to Professor Azbar. Thank you very much, and have a nice, also tomorrow have nice presentations. Thank you very much.
Thank you very much. Goodbye.
Bye.
Thank you, Barış. It was an excellent moderatorship. I really appreciate your help for us. And hi, Roland. It's been really good to see you again after several years. Thank you for your excellent presentations also.
Thank
This
you.
is a very specific issue in Turkey. You know, the poultry sector is a very critical sector in Turkey, and every day the number of the poultries and the chickens are increasing. And in parallel, as you already mentioned, we have huge amount of poultry manure that we have to deal with it. And the nitrogen, as you already mentioned, that it's a big problem because, you know, mainly the poultry sector are willing to have the biogas plants as a monosubstrate. They don't want to have co-digestion with the manure or other stuff.
Yeah.
And it's critical that we have to have some specific technology for this sector. Thank you for your presentation. It was really interesting, and I hope there will be some interest from the audience in coming years. And that was a long day, but I sincerely, I admit that I haven't noticed even a second how fast the time flies today. I was really excited and really enjoyed every single presentation. I'd like to thank to our participants, where we are still 189 people. This is I think, tremendous number for a long day. And I'd like to thank to our honorable and distinguished speakers and the moderators for today. Our next program is to show you some company presentations for those of you who are interested in, and we will have some videos for you. Apart from that, I'd like to say bye for today, and I'd like to see you tomorrow morning at 9:30 for the second round of this workshop. I hope you enjoyed the first day, and I wish you a good day for the rest of the day. See you tomorrow. Bye-bye.
We, the PRE company, stand for Power Recycling Energy Service. We are a small, medium-sized company located in the north of Germany. Located in the north of Germany, near the Baltic Sea. We have been around since 1995, so we have over 25 years of experience. We are an innovative and broad-based company with many years of experience and knowledge in the biogas sector. Our company consists of a team of engineers and scientific employees. We cover the entry spectrum of the biogas industry from planning, development to construction, commissioning and monitoring of projects. In particular, we have been specialized in the production of renewable energies and the use of the biomass. PRE plan project biogas plants, local heating networks, heating, cooling and air conditioning systems. We support and accompany you in all phases of project development and realization. Our company is also very well positioned for the future. PRE has a well-equipped in-house laboratory in its own small research and development department. PRE is able to support their clients in the biological and chemical issues in the biogas sector. In numerous projects in the past, PRE was able to research and develop many of the products that are on the market today. This is how the express hydrolysis was created. With this patent process, we are able to cyclically increase the degradation rate of various feedstocks. This means that the biogas yield can be increased by the separated hydrolysis stage. Another important business area of PRE is the distribution and use of the patented wavebox. This ensures that the fibrous components are dissolved in the substrate and that the biochemical hydrolysis takes place even more efficiently. This means that more methane can be produced from the organic substances. We are happy to help you with personal maintenance and to make your biogas plant even more effective with our products.
Ekorek ile toprağınızı canlandırın. %50 organik madde ve %20 hümik fülvik asit içeriğiyle toprağınızın su tutma kapasitesini artırır ve pH'sını dengeler. Çorak arazilerinizi kısa sürede ıslah eder. Solucan oluşumunu artırarak toprağın havalandırılmasına yardımcı olur. Toprağın ihtiyacı olan besin elementlerinin alımını kolaylaştırır.
Rengini aldıktan sonra kalite varsa, sağlamsa tüccar kesiyor gidiyor. Orada da bir şikayet gelmedikten sonra bu sefer alanı da satan da memnun oluyor. Yani bu şekilde şu anda bayağı raf önünü uzattı, kaliteyi oldu. En çok sevindiğim de 10 ton yapan bir bar şu anda 20 ton yükseldi. Ben kullandım, yüzüm güldü. Diğer arkadaş çiftçilerim de gülmesi için herkese tavsiye ediyor ve ettim.
Ekorek, organik gübre.
Normal klasik şekilde kimyasal gübrelerle bakımımızı yapıyorduk. Ekorek gübreyle tanıştıktan sonra bunun uygulamasını yaptık. Hatta geçen seneden 2 tık fazla verim aldık. Renge de çok etkisi var gübrenin. Renk almasında şeyinde çok etkisi var. Herkese tavsiye ediyorum. Albenisi güzel renk olarak da arttırıyor rengini. Daha beneli yapıyor. Tabii bu müşteriye de yansıyor. Bize de yansıyor.
Ekorek, organik gübre.
Şimdi bu arazide bizim hiçbir şekilde bir ürün alamıyorduk. Yani çorak araziydi burası. Yani öyle bir şey ki terk edilmiş bir arazi gibi bir durumu vardı. Daha sonra Ekorek firmasıyla görüştüğümüzde bunların bir organik gübresi olduğunu, kompost gübre, yani geri dönüşümden elde ettikleri bir kompost gübreyle böyle bu arazide bir deneme yapmak istediler. Biz de kabul ettik ve bu araziyi 2017, 2018 yani 2019'da ektik ürünlerimizi. 2 yıl bir gübrelemeyle bu araziyi bir kontrol altına aldılar. Hem bizim takibimizde hem onların kendi kontrollerinde ve gördük ki 2019'da artık tamamen arazi bizim hiç çorak alan kalmadı. Her yeri yani yeşil otla kaplandı ve sanki tarlanın toprak yapısı %100'ü değişmiş gibi bir durum hissettik.
Ekorek, organik kompost gübre.
Bağımda üzüm yoktu. Biz ziraat mühendisi getirdik. Bizim yazıda traktörü var dediler. Bağı sökmemi söylediler. Abimle tanıştım. O dedi ki böyle ekolojik bir gübre var, organik bir madde var dedi. Bunu bulduk. Şimdi bu kadar masraf ettim, şimdi bunu kullanayım. Az bir miktarda aldım, ton gibi. Yani o denemek için bağın en kötü yerine attım. O sene bizim yaptık. Bunu yaptı. Bu işe 14 yaşında sen ne yaptın dedi. Yaptım bu bağı. Gerçekten faydasını gördüm. Hastalıkta, diriliğinde, bitkinin her aşamada farkını gördüm.
Ekorek, organik kompost gübre.
İlk etapta katı atıkların bertarafı durumu söz konusuydu.
Evet.
Türkiye'de de bu konu özellikle 2000'li yıllarda, hatta ondan da biraz öncesinde çok daha yeniydi. Pek çok şehrimizde bu konuyla ilgili atılmış çok ciddi yatırımlarda bulunmamaktaydı. Dolayısıyla o beni öncelikle bu işe sevk etti. Ama arkasından tabii şunu da hep düşündük. Bu ürünler, organik nitelikli bu ürünler doğaya kazandırılabilir, doğaya faydalı ürün haline de dönüştürülebilir. Zaten mesleğimizin de bir parçası. Dolayısıyla bunlar birbiriyle örtüştü. Katı atığı bertaraf edelim, doğaya zararsız hale gelsin derken güzel de bir ürün haline getirdik. Şimdi onu tekrar gübre olarak doğaya geriye sunuyoruz. Biz burada bitkisel atıkları özellikle kabul ediyoruz. Nedir bunlar? İşte özellikle gıda endüstrisiyle uğraşan fabrikalardan çıkan üretim artıklarını yahut da işte tarihi geçmiş ürünlerin, burada şimdi marka söylemeyeyim ama tarihi geçmiş ürünlerin işte yoğurttu, peynirdi, ondan sonra süt ürünleriydi, patatesli, sebzeydi, meyveydi. Bu tür ürünlerin bizim tesisimize geliyor. Çevre Bakanlığı tarafından lisanslı bir firma olduğumuz için öyle de yapmaları gerektiğinden dolayı bize geliyor. Biz de onları çeşitli işlemlerden geçirdikten sonra asıl ana işlemimiz zaten fermentasyon. Açık alanda fermentasyon işlemi yapıyoruz. Ondan sonra bunu organik gübre haline getiriyoruz.
Ekorek, organik kompost gübre.
Yenilenebilir ve diğer enerji alanlarında gerçekleştirdiği çevreci ve yenilikçi yatırımlarla Türkiye'nin enerji ihtiyacında dışa bağımlılığını azaltmasına katkı sağlayan faaliyetler yürüten Energrom Enerji, atık ve biyogaz alanında araştırma, geliştirme ve uygulama konularında öncü olmuş uluslararası mühendislik ve yatırım firmasıdır. Energrom Enerji, Türkiye'nin artan enerji talebi ihtiyacını karşılamak üzere yenilenebilir enerji santralleri yatırımlarına devam etmeyi hedeflemekte olup, mevcut yatırımlara ek olarak yeni yatırım projeleri üzerine olan çalışmalarını da sürdürmektedir. Biyoteknoloji ile biyogaz üretimi, yarattığı katma değer aracılığıyla hem tarım ve hayvancılık hem de enerji sektörüne önemli katkılar sağlamaktadır. Energrom biyogaz enerji santralleri, yenilenebilir enerji sağlamanın yanı sıra biyolojik kompozisyon açısından zengin sıvı ve katı organik gübre üretmektedir. Bu nedenle biyogaz tesisleri, ekonomi ve çevre açısından bulunduğu bölgeye fayda sağlamaktadır. Ekim 2019 tarihinde faaliyete başlayan İzmir-Foça Biyogaz Enerji Santrali, 3.2 megawatt-saat kurulu güce sahiptir. Tesiste yılda 159.500 ton büyükbaş ve tavuk gübresi fermente edilerek 22.407 megawatt-yıl elektrik üretimi gerçekleşmektedir. Proseste kullanılmakta olan ana ham madde kaynağını bölge çiftliklerden sağlanan büyükbaş ve tavuk gibi hayvansal atıklar oluşturmaktadır. Tesisin organik gübre üretim kapasitesi yaklaşık 13.000 ton bölü yıldır. 2021 yılında YEKNEM kapsamında üretime başlayacak olan Balıkesir Biyogaz Enerji Santrali 3.2 megawatt-saat kurulu güce sahip olacaktır. Ana ham madde kaynağını yine bölge çiftliklerinden sağlanacak büyükbaş ve tavuk gibi hayvansal atıklar oluşturmaktadır. Tesiste yılda 189.800 ton büyükbaş ve tavuk gübresi fermente edilerek 22.407 megawatt-yıl elektrik üretimi gerçekleşecektir. Tesisin organomineral gübre üretim kapasitesi yaklaşık 30.000 ton bölü yıldır. 2021 yılında YEKNEM kapsamında üretime başlayacak olan Aydın Kuyucak Biyogaz Enerji Santrali, 2.1 megawatt-saat kurulu güce sahip olacaktır. Tesiste yılda 131.400 ton büyükbaş ve tavuk gübresi fermente edilerek 14.938 megawatt-yıl elektrik üretimi gerçekleşecektir. Tesisin organik gübre üretim kapasitesi yaklaşık 12.450 ton bölü yıldır. Energrom Enerji ve topluma saygı ilkesi çerçevesinde çalışanları ve iş ortaklarıyla birlikte güven ve huzur içerisinde hedeflerine ulaşma yolunda çevreye ve topluma karşı olan sorumluluklarından ve etik kurallarından ödün vermeden ilerlemeye, gelecek kuşaklara yaşanabilir temiz bir dünya bırakmak için hiç durmadan çalışmaya devam etmektedir.
Ersis Enerji firma tanıtım sunumumuza hoş geldiniz. Ersis Enerji, enerji santrali projelerine yönelik mühendislik, müşavirlik, danışmanlık, proje yönetimi ve proje geliştirme, denetim ve yapım hizmetleri veren bir firma. Firma çalışanları yurt içi ve yurt dışında edindiği deneyimleri Ersis Enerji aracılığıyla piyasaya sunmakta. Bunları yaparken uluslararası proje yönetim ölçütlerini özenle kullanmaktadır. Ersis Enerji bünyesinde biyokütle, biyogaz, kömür, doğalgaz, hidroelektrik, jeotermal, rüzgar ve güneş santrali enerjisine dayalı endüstriyel ve güç üretim tesisleri için fikir oluşturulmasından işletmeye kadar tüm aşamalarda çalışmış, proje geliştirme ve yönetimi, yapım, danışmanlık ve işletmelerde performans iyileştirmeleri üzerine tecrübeli bir ekip, enerji projelerinin her aşaması için hizmet sunmaktadır. Biyokütle, biyogaz, hidroelektrik, jeotermal, güneş, rüzgar gibi yenilenebilir sektörlerde, kömür, doğalgaz, nükleer gibi konvansiyonel sistemlerde, atık su arıtma çamuru, hayvansal atıklar, bitkisel atıklar,
conventional side,
şehir
we
çöpleri
have
gibi
coal, natural
atık
gas,
bertarafına
electricity
yönelik
here.
tüm sektörlerde hizmet veren
We
bir
are
firmayız.
working in all sectors in terms of waste management.
Santral projelerine yönelik fizibilite çalışmaları, izin süreçleri yönetimi,
Feasibility studies
mühendislik,
and
tasarım,
permit process
teknoloji seçimi,
management, engineering,
inşaat,
design,
montaj,
technology selection,
denetim işleri yönetimi,
construction,
devreye
assembly,
alma,
and audit
işletme ve
management.
bakım temel hizmet alanlarımız.
Business and maintenance are also our
Verdiğimiz
main areas of
hizmetler
expertise.
santral ön
Our
fizibilite ve
services
fizibilite
consist
çalışmaları,
of
mevcut durum
power plant
tespiti, yatırım danışmanlığı,
primary feasibility,
temel ve detay
feasibility and
mühendislik hizmetleri,
feasibility studies,
proje ve tesis
uh,
konsepti
due
geliştirme,
diligence,
ihale şartnamelerinin hazırlanması ve ihale süreci yönetimi, lenders
tender
engineering
process
hizmetleri,
management, lenders
sözleşme
engineering,
danışmanlığı, yapım süreci proje yönetimi,
consultancy,
performans
agreement
değerlendirme ve
contract,
iyileştirme danışmanlığı,
project
rehabilitasyon
management,
analizi ve yönetimi,
construction process,
teknoloji seçimi,
performance evaluation
kontrol
and
ve
procurement
denetim olarak
consultancy,
başlıca
rehabilitation analysis,
sıralandırılabilir.
technology selection, control and audit.
Devam eden bazı referanslarımızı hızlı
have
geçmek
some
adına
of our references here.
biyokütle santrali projelerimiz, biyokütle santraline yönelik
We have
santral ve
biomass
tedarik
energy
zincirinin
plants
kurulmasına yönelik fizibilite
also
hizmetlerimiz,
works and
Akkuyu
feasibility
nükleer
works
santrali
for
inşaat dönemi denetim
some of
hizmetleri,
our projects, some
hizmetleri
audit
işimiz.
projects as well.
Yine biyokütle santrallerine, yakma tesislerine, biyogaz enerji santrallerine yönelik arıtma çamuru yakan
We can
sistemlere, çöp gazı
we
ve
have
biyogaz
systems
santrallerine
that
yönelik
use
danışmanlık, proje yönetimi
wastewater and
ve
slag.
deprem mühendisliği kapsamında yaptığımız
We
işler var.
provide
Tamamlanan bazı projelerimizi hızlı geçmek istersek, rüzgar enerji
if we
santraline
want to talk
yönelik,
about some of our
firmaların
completed
kazan sistemlerine yönelik,
projects, you can
gazı
see some of our references on the screen.
ve biyokütle
Biogas
santrali başta
plant,
olmak üzere
biomass
çeşitli jeotermal
plants as well.
santraller,
Some geothermal plants,
atık bertaraf tesisleri ne yönelik
again some waste
danışmanlık ve
management and
müşavirlik hizmetlerimiz
disposal
devam ediyor.
services.
Bunun yarı sıra yaptığımız ön
Along with
araştırmalar,
these,
fizibilite
we
çalışmaları,
have
proje denetim,
feasibility studies, project
özelleştirme
audits,
fizibiliteleri, enerji verimliliği çalışmaları
privatization
devam ediyor.
feasibilities, energy efficiency studies can be listed among our projects.
Tüm projelerimizde uluslararası proje yönetim
Certifications,
standartlarını uyguladığımız gibi
we are
kalite
complying
ve
with
çevre yönetimi
international
açısından
standards
da uygun
and also in
sertifikalar
terms of
sayesinde
environment management, we are
yönettiğimiz
complying with
projelerde
international
tüm
standards.
kalite ve yönetim sistemi sertifikasyonlarımızı,
All
ölçütlerimizi
quality and
kullanmaktayız.
management system certifications
Ilave
are provided.
sorularınız ve yorumlarınız, görüşleriniz
If you
için
have any
bizimle her
further
zaman
questions
iletişime
or any
geçebilirsiniz.
feedback or comments, you can always
Sosyal
contact
medya
us directly
hesaplarımız
through the addresses
ekranda
we provide
gördüğünüz
here.
gibidir.
Our social media accounts are
Projelerimiz
also
ve
visible on
referanslarımız
the screen.
açısından ilginizi
In
çekebilecek
terms of our projects and
projeler
references,
için takip edebilir,
if you have
yorumlarınızı
any
yapabilirsiniz.
interest, you can go to our social media
Sağlıklı günler
accounts.
dileriz.
We wish you a happy and healthy days. Thank you.
You are watching Renesco Energy Company's presentation. Renesco is an undertaking and consulting energy company. We develop technologies on biogas industry with energy efficiency solutions. Renesco is founded by Başar Beyazoğlu in 2017. Mr. Beyazoğlu identifies himself as an entrepreneur and new business developer. Despite of his young age, he has achieved much in his career. He started as a European Union project writer. With his success, he gained the right to full scholarship education in England. Then, he established his first company in energy sector. Later, he continued his career in UK with Hydromix International. Right now, Mr. Beyazoğlu continues his work to develop Renesco as an international leading company. As Renesco Energy, we are specialized in these topics: anaerobic digestion, wet and dry, municipal waste and landfill, mechanical biological treatments, solid and liquid fertilizers. With our experienced team, we have the responsibility of designing and building our biogas plants. After the construction of a new facility, we operating it with the most efficient way with the power we get from our other facilities. We use our experiences to develop our business and carry it further. Maintenance also is a very important subject that Renesco always gives importance. Through our wide supply chain, necessary interventions are made on time. Having many plants helps preventing efficiency losses. Renesco Energy always updates its knowledge by creating business partnerships. We put together latest technologies and developments in our plants. At Renesco, we always work with best qualified companies. Creating good relations with our suppliers is important for us. We chose bulk purchasing to able to give most optimal price for our customers. Tracking of plants have much importance in Renesco Energy Company. From our headquarters, we watch all our facilities to keep efficiency at maximum level. By doing this, potential issues can be corrected in advance. Renesco Energy is also the first and only member of World Biogas Association from Turkey. We continue growing in biogas sector by creating new partnerships. Our transparent management manner provides profitability to our partners. Renesco Energy is a huge family, and it keeps growing with new facilities. Currently, we have 11 installed power plants and 2 installed EPC projects which have capacity of 32 megawatts. By the end of 2021, Renesco Energy aims to reach 80 megawatt capacity. Thank you for your attention.